zal in Nederlands dus maar voor hier uit te praten.
Gewoon daarop. Gewoon erop leggen.
Do you know what's the other studio?
No. I'm just opening it.
I just see you speak with team chat.
No, yesterday I followed the jury.
Okay.
So we have first one, Michelle is the first one. Yeah. Then you have second one, Katrina, the one not sure. Yeah. And you have also the PDF, which you also.
Perfect. Then the guys can do what they want. Team.
We are ready to begin. Welcome. Session 1 of the first day of this thesis presentation. We will have 3 students present their thesis, and you will have 15 minutes to present your thesis, and then you will have 15 minutes for questions. First, we will go through the questions from your jury, which I will present in a minute, and then if there's time, we're going to ask if the public here attending in presence has questions, and then if the online chat has questions. So also, hi everyone who's following online. And I will start presenting the jury. So we have Professor Silvia Bianchelli from the Polytechnic University of Marche. Then we have Dr. Heidi Acampora from the ATU University in Galway. And then we have Professor Gonzalo Machado Schiaffino from the University of Oviedo. So the first to present is going to be Michelle Pingidore, so you can approach. Michelle will talk about sea turtles, but I will let you introduce your title by yourself. Please just be sure you don't close the software of the YouTube live. Okay, and you can start.
Good morning, everyone. I'm Michelle Pingidore, and today I'm presenting my thesis evaluating the influence of biologging devices on sea turtles. In the last decade, animal-borne devices, here referred to as biologgers, have revolutionized the study of marine megafauna. Biologgers provide important insight into the spatial distribution, behavior, and surrounding environmental parameters of marine animals, data that would otherwise be difficult to gather. Since their increased usage in marine science, it is also important to know which are the potential effects of these devices in endangered species such as sea turtles. We identified issues related with bilogger attachment and bilogger retention. I don't know if you know, but in sea turtle, biloggers are deployed on the carapace, and the protocols involve mainly two factors: animal handling and interaction, which itself could cause stress to the animal, and the use of adhesive epoxy glue. Epoxy glue comes into two components, and when these two components mix, they undergo an exothermic reaction that releases heat in the surrounding surface. Concerning biologger retention, which is basically the presence itself of the device on the marine animal, this can disrupt animal hydrodynamics and buoyancy, thus leading to changes in swimming activity, predation success, predator avoidance, and in general increased energy expenditure. In this study performed at the Oceanographic of Valencia, we aim to evaluate the short-term effect of both bilogger attachment and bilogger retention. So concerning bilogger attachment, we developed two different research questions: how epoxy glue affects body temperature of loggerhead turtles, and how bilogger attachment protocol alters heartbeat fluctuation again on loggerhead turtles. To evaluate instead the effect of bilogger retention on sea turtle, we plan an experiment to focus on behavioral disruption on both loggerhead and green turtles held in captivity at the oceanography. Now I will show you in detail the methodology of each experiment, and later I will show you the results. So for the first experiment, we focused on understanding the thermal effect of epoxy glue on sea turtle carapaces. For this experiment, the sample size were 4 carapaces of naturally deceased loggerhead turtle, and we applied 4 different types of epoxy glue on the second and third lateral scutes of the carapace of sea turtle, as you can see in the picture. The choice of the 4 types of epoxy glue is just random choice based on some of the most common type of epoxy that scientists use in marine conservation. We basically measured the temperature of both external and internal side of the carapace with an infrared thermogun, starting from time 0 to have an initial thermal state of the turtle, then every 2 minutes until epoxy curing time, which is basically the time at which the epoxy solidifies, so the exothermic reaction is completed, and then every 5 minutes for a total of 60 minutes. To evaluate the physiological response of loggerhead turtle during bilogger attachment, we monitor heartbeat fluctuation on a loggerhead turtle. The Oceanographic of Valencia was deploying a satellite transmitter on this turtle, so we basically opportunistically choose it as our sample size. And the experiment was divided in 3 different phases: the control phase, which didn't include any animal handling; the bilogger attachment phase, in which we deployed the satellite transmitter on the turtle; and then the recovery phase, in which again there was no handling of the animal. For the behavioral study, instead, our sample size was 10 individuals of loggerhead turtle that were housed in the rehabilitation center of the Oceanographic of Valencia, and 7 individuals of green turtle that were instead permanent residents of the aquarium installation. This study was also divided in 3 different phases, actually 3 consecutive days. The first day being the control, in which there was no handling of the animal, and I just observed the animal for 4 hours. Second day, bilogger attachment day. We personally deployed the device that you see here. It's called Turtle Cam, using a fast-setting epoxy glue, and then again observed the animal for 4 hours. And the last day is the bilogger detachment day, in which we removed the device and the epoxy with a small spatula, and again observed the animal for 4 hours. We included this last condition because with this we were able to separate between animal handling related stress and stress related solely on bilogger retention. Due to practical matters, the behavior of loggerhead turtle was recorded with a video camera and then analyzed with Boris software, which is a software specified in etograms and video analysis, while the green turtle behavior was basically directly annotated by me through live observations. This is a small representation of what the ethogram that I used looked like. So we had 6 main categories: swimming, resting, scratching, eating, breathing, and other, with a 7th one, interactive, that only applied to green turtles, because in each aquarium installation there was a minimum of 2 green turtles individual, while for the loghead turtle, as you can see in the picture above, in the rehabilitation center they were held in solitary for health purposes. I would like also to put a focus on the scratching behavior. Basically, scratching was annotated when the turtle was moving the body back and forward against artificial rocks and corals present in the aquarium installation, or against brushing tools that you can see again in the top picture that I built myself, that are basically environmental enrichment devices. Talking about the results, evaluating the thermal effect of epoxy on loggerhead turtle carapace resulted in a general external and internal carapace temperature. As we can see from the graphs, the external carapace temperature was higher than the internal one, and as a general rule, the highest temperature peak was recorded around within 5 minutes after curing time for each epoxy. When instead we were evaluating the physiological response of bilogger attachment on loggerhead turtle, we saw an increase of heart rate by 5 beats per minute during bilogger attachment compared to the control, and a subsequent decrease of 19 BPM during the recovery phase. We also performed statistical analysis on this, and it was revealed that there was significant difference of heart rate values among all the 3 different conditions. Comparing the behavior of 10 loggerhead turtle resulted in increased swimming and decreased resting behavior in both device and no device day, while behaviors such as scratching, eating, breathing, and other were only depicted less than 2% of the time along the different conditions. Statistical analysis reveals significance in swimming and resting, and we perform a one-way ANOVA or Kruskal-Wallis test for non-normally distributed percentage data. When instead comparing the behaviors of 7 green turtles among the different conditions, it resulted in a decrease in swimming, as you can see from this graph, and an increase in resting. Behavioral categories such as scratching and interacting were more pronounced during the device day, while I want to put a focus now on the category of other, which basically included one peculiar behavior. The turtle was trying to remove the device using the front flipper. I have a video after that will explain better. Statistical analysis revealed significance in swimming, resting, stretching, interacting, and other, again using one-way ANOVA or Kruskal-Wallis for non-normally distributed data. And here I have the 3 videos. So in the first video you can see how a green turtle scratches the carapace against artificial corals, trying to remove off the device. In the second video we clearly see 2 turtles interacting. Both of them have the device deployed, and one is trying to bite it off, basically. And for the third video, I kindly ask you to pay attention to the right front flipper, because this turtle will do this peculiar movement that I told you 3 times. Now it's just swimming, and then... So, to summarize, the setup experiment conducted during this study aimed to focus on the short-term effect of both bilogger attachment and retention on sea turtle. Our result explained that epoxy glue application increases internal carapace temperature of 1.6 to 4.5 degrees. This is just an overestimation. We do not know the actual physiological effect on sea turtle, as these experiments was performed on dead carapaces. And also we saw that physiological factors such as heartbeat fluctuation increase during bilogger attachment protocol. So this means that the protocol itself causes stress to the animal. Apart from physiological alteration, we also discovered that bilogger retention influences sea turtle behavior. With loggerhead turtle, the behavior was more actually influenced by the handling itself and the bilogger attachment procedure, because as we saw in the previous graph, both device and no device day were mimicking the same response, and also we had little minimal scratching behavior, so this means that bilogger retention was not perceived as a stressful factor. On the other hand, greedy turtle swim less during device and no device, and rested more during the experimental period, and this is also related to animal handling related stress. But as we saw, with high scratching activity and that peculiar movement of the turtle trying to remove the device with the front flipper, we can understand that here the bilogger itself it was perceived as a stressful factor. The differences between responses of different species may be due to species-specific morphological trait, general tolerance to epiviolence, and also general ecological behavioral pattern. So, what should we do next? Future studies should look at the long-term impact of both bilogger attachment and retention on sea turtle, focusing on intraspecific responses. Scientists also need to be conscious on animal handling related stress, and there is the need of developing new protocols and less invasive bilogger devices, especially in sensitive species such as green turtles. These studies are not only needed to minimize stress and ensure animal welfare, but also to improve data reliability and minimize behavioral disruption in wild animals. Now, if I have time, I would like to show you a brief video that just summarizes the bilogger attachment procedure. Thank you.
And now I would like to leave the word to the jury. We can start with Celia and Kelly. I just want to remind you not to go in between sessions between the
Hi, Michelle. Thank you very much for your presentation. It's a pleasure to hear all of your work done during this year. I have several questions, but you already answered my major questions during your discussion and conclusion. So I have just a couple of curiosity. First one, if you think that 4 hours are sufficient to give, complete observation, and I wonder if waiting for a longer time could leave the animals somehow adapting to this device attached.
Yeah. So previous study basically focused on looking at the behavior for 2 hours. So our goal was to increase this observational period, but due to time concern, we were able only to extend it for 4 hours. But seeing the results, I think we do need to extend the observational period maybe to 24 hours, so we can have a general overview of the turtle behavior. Also, I don't know if it was clear from the presentation, the biologger stayed on the turtle for 24 hours. But we don't know what happened after I stopped the observational period. So yeah, I agree.
Okay, this could be further
Yes.
another study. And second question is if there are any information from the natural environment. I don't know, with satellites, information deriving from the radios, bivalves.
So you mean which type of information we can get?
About the changing movements in the behavior of animals.
So
Because I
Yeah.
mainly need to explain myself better. You did, in my opinion, you did a very complete job analyzing a lot of different characteristics of the behavior of animals. I think that from natural environment, this study would be very difficult to do. But I wonder if there are any information or observation of same or similar information in the natural environment.
So there are actual,
Previous studies.
There are previous studies that already discovered, especially on green turtles, that they tend to scratch more and also have aggressive behavior like biting between each other. And this is reflected also in the captive study. But normally when scientists do these behavioral studies in the wild, they either use the turtle cam, which is basically a camera, so we are able to record the behavior. Since the camera is on top of the carapace, we see what the turtle is seeing. But again, the perspective is different if we compare it to like us observing directly the animal. And also scientists are using drones, which are also useful, but again, we kind of miss some type of behavior, especially as the turtle goes deeper in the water.
Okay, thank you. So we can go to
Hi, Michelle. Thanks very much and well done on your presentation. Looks like really good. I know you had a bit of a small sample size, especially on the harpies, and I wanted to ask you why you only did one turtle there, since you actually were also handling the other ones for the behavior.
Yeah, because I can show you the picture. So, as you can see here, the whole satellite transmitter was deployed, and this is like the basic protocol that scientists normally use. And we didn't want to deploy satellite transmitter if it was necessary on turtles, since we saw that it was causing stress. And I couldn't do the same type of experiment in these ones. As you can see, the amount of epoxy applied is completely different from this type of biologging devices. So this was the main reason. And because the oceanographic was deploying only one satellite transmitter during the time that I was there, we were able just to do one sample size.
Okay. And why would you use the ECG? Could you elaborate, like, I mean, you're measuring the harpies, and why are you going through the carapace instead of the classroom?
Well, we basically saw in previous study that the signal is working anyway, so it's easier in this way, and also if you have to handle the turtle and kind of put it upside down, it's causing more stress, and also you can kind of disrupt the organs. So in this way, we didn't really affect the turtle by placing the patches, and the signal was perfect.
Okay, yeah, because depending on the species of the turtle, that kind of it wouldn't be very efficient. But you did obviously
Yeah.
test
Also
that
because
of the carapace
the-
being thicker or not.
the individual had to be minimum 10 kilos, and this was 60 kilo individual. So that's why we had a strong signal.
And just one last question. So in your discussion there, you said that loggerheads swam more due to handling, and then you also said green turtles were resting more due to handling
Yes.
as well. So, like, how would you be able to like define that this was the cause in terms of? I know behavior studies, you know, they can be a bit subjective in that sense.
So basically, by looking at this proportion of time spent, we saw that during device and no device, the only common thing was the animal handling. So if the stress was only strictly related to biological retention, we would expect the no device day to basically be similar to the control and not decrease. As you can see, the swimming activity is even decreasing more. So our thought is that probably the animal didn't recover from the handling stress of the previous day, and since we took it out again from the water, it gets even more stress, and this could explain why the behavioral allocation.
Okay, and could you just a last question, I promise. Could you just explain to me again how you did the control handling?
Basically, I was just observing the animal for 4 hours. We always started at the same hour every day, so all the day had the similar environmental condition. Especially here, since it was in the aquarium installation, I personally asked not to dive, not to do like other procedures, because I needed the 3 days to be equals. So yeah, basically I just observed the animal with my computer, with the body software, and annotated the behaviors. Thanks.
Okay, thank you. Moving on.
Okay, so thanks a lot, Michelle. Thank you for such a nice piece of work. I think that I didn't have the chance to read your written manuscript, but I will start with highlighting the relevance of your work, yeah, because I think it's a really nice experiment, set of experiments that are trying to analyze the effect of biologics and why biologics are so important for knowing many important biological characteristics of certain species that are very difficult to monitor. So in that sense, I would like to highlight even more your thesis.
Thanks.
I mean, this is not my expertise, but in any case, related with Heidi just said, which is every time that they are handling, it doesn't matter if you are adding biology, which is the change in heart beats. Do you know that? Do you have that?
No, we didn't look at that piece of information, but I actually think it's...
Because it would be nice just to tease apart which part of an movement is just due to normal handling and which is due to the addition of these biologics.
What I can say is, if you look at the red line,
yeah.
So basically, here, there was no more handling of the animal, but since the exothermic reaction of the epoxy glue was still going on, it still counted as biologger attachment procedure, and we see that as soon as we stop being around the animal, the heartbeat increases. So, for sure, animal handling is probably the main stressful factor here. We couldn't differentiate between the epoxy glue effect on the turtle or just us being around the animal, but yeah, I think this could be a starting point.
Okay, so that's great. And related to the graph that you show us, the difference between the bilateral device and the control.
Yeah.
So these are the same individuals, yeah? You're talking about the same individuals?
Yes, this is an average of 10 individuals. So each individual had its own response because each individual has its own personality.
Yeah.
But as an average, they all responded in this way, and also in green turtles, all the individuals scratched more, all the individuals did this peculiar movement with the front flipper. So we can say that even if the sample size is not that big, we are pretty positive that this is the actual response.
Because how do you think to about analyzing by since you have the same individuals with control before and after, right? If I'm wrong.
Yeah, it's the same individual
So to see
3
the
times.
change per individual instead of just pulling the as a group.
I mean, yeah, but we also, at the beginning, we also wanted to see if there was change related to sex and change related to size. But because for some of the turtle we didn't know the sex and for the size we didn't see much difference, like they tend to behave the same, that's why we decided to just show the general graph.
Okay, that's it. Thank you very much.
Thank
Good
you.
job.
Okay. Thank you to the jury members. And now still in time, if you have some questions, we have around 3 minutes. So, 1, 2, 3. Okay, so Catherine.
Thank you so much for your presentation. This was really interesting. I'm just wondering where your turtles came from, if you collected them from the wild or if they were already living in an aquarium.
Yeah, so the loggerhead turtle that were in the rehabilitation center, they were basically bycatch by fishery. So yeah, they came into the sonography to be treated and later released in the wild. Again, we opportunistically used them before release them back in the wild. While the loggerhead turtle were permanent resident of the aquarium, I know that they arrived in the sonographic around 3-5 years ago, and they're probably coming from other aquariums. So I don't know like their whole background, but I'm sure they're captive animals.
Yeah, I'm just curious with the amount of toxic material that you use. In one of the videos, I saw that you added, they have like the device looks like in the middle is very big, and then on these like little branches that it has.
Yeah.
I'm just curious, why did you choose, like did you have to use a specific amount of this, like how did you control that you didn't add more of this glue in one turtle than in the other?
Well, basically, I don't know if you can see from the picture, but there is a small plastic mesh of 4 times 3 centimeters. So the epoxy glue, the amount of epoxy glue used was basically, you had to do a thin layer and then put the plastic mesh on top and cover it again with epoxy glue. And I don't know if it was clear, but there are 3 different attachment points. There is one here, one on the other side, and then the device, as you said, is pretty big. And there is another little plastic mesh at behind the device to give stability to the whole transmitter.
I actually have 2 small questions. The first one is, do you know the lifespan is of the biologger in case of battery
Yeah,
generation?
so it depends on the type of device. We have different ones. This specifically lasted 6 months, but then you could put a SD card to actually record the behavior. So for this specific one, you could, to gather the data, of course, you had to go and track the turtle, take it back and take off the SD card. So then you could remove it. But normal GPS devices, as the one that I deploy on for the heartbeat measurement, they last 6 months, and normally they have an internal technological setup that it's kind of like pulling out the device once the battery is gone, so the turtle is free again.
We actually don't have any more time for questions, but you can ask her after the session. And yeah, thank you again.
Thank you.
And now from marine turtles, we move to invasive bivalves with the presentation by Catherine Moore. So please go ahead now for the presentation.
Good morning, everyone. Thank you so much for being here. My name is Catherine, and today I'll be talking about connectivity of invasive bivalves between Galicia and Brias. This is one of the study species. It's the invasive mussel, Xenostrobus securus. To first contextualize the problem of invasive species, 60% of biodiversity loss is attributed to the spread of invasive species, and this cost a yearly amount estimated to be $423 billion, 10% of which is coming from marine invasions. These numbers are coming from the Intergovernmental Panel on Biodiversity and Ecosystem Services 2023 report. Target 6 of the Global Biodiversity Framework states that identifying pathways of invasion is crucial information for management, so that is what I aim to do in my study. The Galician Brias are these long, narrow inlets, and they are very productive ecosystems because there's a river that flows into the head of the Bria, and there is marine influence here at the mouth of the Bria. Because of the ecologically rich conditions, these are a prime site for aquaculture, which you can see pictured here. There are two species that are the focus of this study. They are the invasive oyster, Mygalana gigas, which is pictured here as the white oyster, and the invasive mussel, Xenostrobus securus, which it's mixed in with algae, but you can see it here. And these species pose multiple different threats. They have the potential to expand and colonize large areas. This is further enabled by their pelagic larvae stage, where they spend 3 to 4 weeks dispersing with currents, allowing them to colonize new environments. They also can be a vector for pathogens and other invasive species, which has the potential to inhibit aquaculture, which is very important to the region. And both of these species are very adaptable to varying environmental conditions. So given the context of the study species and the region, the research questions that this thesis addresses is, where are new recruits of these species coming from? Are populations between Brias connected? And if so, which subpopulation is acting more as a source? And which environmental and hydrodynamic factors are facilitating dispersal of these species? So, the method that this study uses is an empirical method called geochemical tags, also called elemental fingerprints. And the reason this method is selected is because traditionally connectivity studies use biophysical models. However, often these biophysical models require empirical data to validate them. So this provides empirical data about population connectivity. And the way this works is that both within and between Brias, there are different water compositions. You can see some factors here that might impact the water conditions, the water composition. This includes runoff from rivers, runoff from land, proximity to development, as well as other things. And then when these bivalves start to form their carbonate shells in the first 3 days of their lives, they uptake these elements in the water at a rate proportional to environmental conditions. They then spend 3 to 4 weeks in their pelagic larvae stage where they disperse, and then they settle, but they actually retain their geochemical tag that they obtained at their natal site. So if you see here, this is a larvae, and this is a recruit, and you can actually see the part of the recruit shell that was obtained at the natal origin. With this in mind, here's the sampling design for this study. So in the summer, wild adults were collected and spawned in the lab, and then larvae were placed into larvae homes that were incubated at each study site. They were kept there for about 5 days, and then they were removed and frozen until processing. 1 month later, recruits were collected using these scattering plates, which you can see on that picture. I apologize for it being so small. And then they were also frozen until preparation for laser ablation inductively coupled plasma mass spectrometry, abbreviated LAICPMS, which I'll talk about more in the next slide. All of these sites were the oyster sampling sites. However, recruits and larvae were only collected from the northern and central sites, which is in line with where the population has naturalized. I prepared larvae and recruit samples from each of these sites. However, there was equipment failure, so these samples were unable to be processed in the time frame of this study. So the rest of this study will focus on mussels, which are another invasive species. And these are the 2 Brias where the mussels have already naturalized. They are Rio de Vigo and Rio de Pontevedra. They are both in the south of Galicia. Larvae were incubated in these 2 sites at Pirguas and Ponte Sampaio, in July 2017. And then recruits were collected from these 4 sites, Clares, Paraguas, Ponte Sampaio, and Santa Cristina, 1 month later in August 2017. However, no recruits were found at Santa Cristina, so that site is excluded from the rest of the study. After preparation, which included cleansing the samples of organic matter and mounting them to a slide, they were analyzed using LAS and PMS, which I'm not going to say again, it's pretty long, but basically how it works is a laser hits the umbo of the shell, which is the oldest part of the shell that developed at the natal origin site, and then the elements in the shell return to particles, which are detected, ionized and detected by a mass spectrometer and detector. After this, I have raw data, which is in counts, and this data was processed using Python. This is what the raw data looks like. First, it is despiked and smoothed to remove unrealistic outliers, as well as to account for noise from the instrumentation. And then the signal region, shown in red, which is where the laser is actually ablating the sample, is separated from the background region in gray. After this, the signal region counts are calibrated using known standard reference materials to obtain molar ratios for each analyte. And so after data processing, I have the molar ratios of 13 analytes of interest, molar ratios with calcium, in every recruit and larvae sample. And then this data was analyzed to assess connectivity pathways and try to determine where the new recruits were coming from. The first thing that I looked at was if the elemental fingerprints were different between the 2 incubation locations, which were in Rio de Vigo and Rio de Pontevedra. And there were 3 elements that were significantly different between these 2 incubation locations. There are cerium, copper, and lead. All of these molar ratios were significantly higher in larvae that were incubated in Paraguas in Rio de Pontevedra compared to larvae incubated in Ponte Sampaio. Next, I wanted to understand which recruit samples had similarities in their elemental fingerprint. So this was done using an unsupervised random forest model. I can talk more about this in the questions if you're curious. But you can see here the clusters. This basically builds 1,000 trees and groups samples based on similarities in their elemental fingerprint. The clusters represent predicted natal origin. It's better visualized here. These are the 3 recruit collection sites. And the colors show the proportion within each collection site that were assigned to each cluster. So you can see that Ponte Sampaio is mostly recruits from cluster 2. Lares is mostly recruits from cluster 3, and Paraguas is a mix mostly of recruits from cluster 1 and cluster 2. Then I wanted to understand the elements that were important in differentiating between these clusters. This was first looked at by the mean decrease in Gini index, which shows the most important elements in separating recruits into their clusters. And these are magnesium, barium, lead, cerium, and manganese. And then multidimensional scaling was used to visualize the separation of clusters based on their similarities, and the molar ratios were projected onto the MDS. So what we can see here is that cerium, manganese, and barium are all relatively higher in samples grouped into cluster 1. Manganese is relatively higher in samples clustered into 1 and 3, and lead is relatively higher in samples clustered into 1 and 2. So then I wanted to understand the differences in the water composition, both between the 2 RIAs that were the study sites and within each RIA. First, I looked at barium, magnesium, and manganese, and a lot of this information came from literature review that was synthesized with my data. So these 3 elements are all related to weathering of rocks and are higher in areas with higher river input. The average river flow into Rio de Pontevedra is higher than the average water flow into Rio de Vigo. And you can actually see the river here flowing into Rio de Pontevedra, whereas you can't really see it flowing into Rio de Vigo. If you look here, these are the molar ratios between the clusters, and cluster 2 has significantly lower levels of each of these 3 elements compared to cluster 1 and 3. This leads to the prediction that cluster 2 represents an origin in Rio de Vigo, which has lower river input. Next, I looked at cerium. So going back to the larvae data, larvae incubated in Paraguas and Rio de Pontevedra had significantly higher levels of cerium compared to larvae incubated in Rio de Vigo. Also, past studies that have used LAICPMS to assess natal origin of other bivalves have found that those samples have higher levels of cerium if they were born in Rio de Pontevedra compared to Rio de Vigo. And then what I saw is that cluster 2 had the lowest levels of cerium, recruits in cluster 2 had the lowest cerium levels, cluster 1 had the highest cerium levels, cluster 3 also had pretty high cerium levels. So this further supports the prediction that cluster 2 represents origin in Rio de Vigo, cluster 1 and 3 represent origin in Rio de Pontevedra. And then I wanted to try to differentiate between clusters 1 and 3, so I looked at copper. Copper is related to areas with higher maritime activity because it mostly comes from anti-fouling paints. And if you can actually see here, there is a city at the head of the Rio, and then the water from the river brings runoff from the city to the Piriguas site. So it is expected that recruits coming from Piriguas would have relatively higher levels of copper. Also, larvae incubated in Piriguas had significantly higher levels of copper than larvae incubated in Ponte Sampaio. And past studies comparing environmental conditions in water, sediment, and native mussel tissue have all found higher levels of copper in Rio de Pontevedra compared to Rio de Vigo. So this leads to the prediction that cluster 1 likely represents recruits that are coming from Piriguas, which again is over there. All of this leads to the conclusions and main results of this study, which include the cluster origin predictions, being that cluster 1 comes from Piriguas, cluster 2 samples come from Ponte Sampaio, and cluster 3 come from Lares. This was used to build a connectivity matrix, which is shown in the table here and visualized here. And basically what this shows is within each recruit collection site, the proportion of individuals that were predicted to have been born there versus immigrated to that site. And what we can see is that Ponte Sampaio seems to be the greatest source population. Ponte Sampaio and Lares both have relatively higher levels of self-recruitment, which makes sense because they are the innermost sites. And Piriguas seems to be the greatest mixing site. Next, I wanted to understand water movement both within and between the Rias to try to understand the hydrodynamic forces that might be impacting and enabling dispersal. So during upwelling, these are very simplified, we can come back to this, but during upwelling, which tends to occur in the spring and summer, there is southward winds, northerly, if anyone's an oceanographer, and due to Ekman and Coriolis force, the net water movement is offshore. And then within the Ria, cold, nutrient-dense water comes up from the bottom, circulates out, drawing surface water out of the Rias. This is actually the force that enables larvae to move outside of the Ria and then be dispersed to other Rias. During downwelling, the wind blows northward. This is also average water movement due to the poleward currents, and net water movement is into the Rias. And then there's this reverse estuarine circulation pattern where surface water flows into the Rias. During this study period, you see one peak in upwelling here and one peak here, followed by periods of downwelling and upwelling relaxation. And these peaks of upwelling are likely what allowed the larvae to leave the Rias, and then the downwelling and upwelling relaxation is what facilitated that northward expansion, which is why we saw greater mobility from Rio de Vigo to Rio de Pontevedra. So, to summarize and bring it all together, the greatest source population was determined to be Ponte Sampaio. This is enabled by some biotic factors as well, such as the largest population density and largest individuals at this site, leading to higher gametic production capacity, and warmer temperatures in this Rio, which enhance spawning. There are variable climate models for this region, but it's very important to understand what might happen in different climate scenarios. So in a reduced upwelling scenario, there will likely be higher self-recruitment because reduced upwelling and downwelling do lead to greater circulation within the Rio. In an increased upwelling intensity and frequency scenario, there might be southward expansion and Pontevedra might start to act more as a source. This all leads us to recommendations for management. There should be monitoring of the aquaculture ropes where the native mussels are growing, just in case the species continues to expand further into the Rias, because then they could inhibit aquaculture. And also the northward sites, which is the Rio da Rosa, should be monitored for further expansion. And one way to manage this population is by engaging on-foot shellfishers who are losing their livelihoods. Future research could focus on more comprehensive connectivity matrices. And I think that was a lot of information. Thank you so much for your time and attention, and I'm really happy to answer any questions you have.
Thank you for your super nice presentation. You were super clear. I have just a suggestion for you. Please make the graphs bigger next time, because from I don't know, and the words were sometimes too little to be read. I have just a couple of questions in terms of the approach that you use. Do you think that this approach could be also applied to other species?
Yes, so this geochemical tag and elemental fingerprinting approach was mostly used originally for otoliths to understand fish connectivity, and now it's being used for bivalves. I think that anything that has a carbonate structure could be used for this technique.
And the second question is, how you mentioned, regarding the management, future management, why did you mention only the aquaculture infrastructures and not the source of the invasion? I mean, such as we could imagine some treatment for waters, I don't know, some monitoring and warning systems.
That is a really great question. So a few thoughts that I have on that. Right now, this species is not interfering with the native species. However, with the invasive oyster that originally was going to be the focus of this study, when it was first introduced to the Galician Brias, it was not believed to be a problem. However, over a few years, it expanded so rapidly that now it is interfering with the native oyster. So there is the fear that this would happen in the future. However, harvesting this species on foot or using other management techniques is time consuming and requires a lot of funding. So that's why one idea is engaging on-foot shellfishers who are losing their livelihoods. But then the reason that aquaculture is relevant is that there are two ways that the native mussel is put into aquaculture systems. There are larvae collection systems, and then wild seed are collected from the field. And the wild seed of the native mussel has been decreasing in the past few years. So there's the potential that if this species expands outward, it could both compete for space with the native mussel, which would further reduce seed stock, and it could settle on the aquaculture ropes. But I think that for now, my goal was to identify priority locations for management, just because looking at locations and monitoring is a little bit cheaper than actually paying people to remove the species, but then provide some recommendations for if the situation gets worse, what we could do to remove the source populations.
I mean, the removal could be most. Thank you very much.
Thank you.
Hi, Catherine. Thank you very much for your presentation. Very insightful. It was a lot of information. Just wanted to put it on that all the first 15 minutes. So I was just wondering, because when I first looked at your abstract, so I was not your thesis reader, but I was looking at your abstract, you said that the recruit mixing aligned with the estuarine circulation. So I feel like, so your results are pretty predictable, I suppose.
Yes, they are, which I think is important because it means that in the future we don't need to repeat this type of study very often, because now we see that the populations are connected between RIAs. There is this movement between RIAs. The alternative would be that they were separate introductions into these two RIAs, but we see that that is likely not the case. And so because we now have this empirical evidence, we can, moving forward, rely more on hydrodynamic and biophysical models, since we already have the empirical data to validate it.
Okay. So what would be your critical insights, like from what you did, even though you thought that was the same thing you would have predicted?
My main critical insight is that the technique is very time consuming. With the larvae, there was a smaller sample size, and the larvae are so small that when the laser ablates them, the signal region is very small and very noisy, and in some samples was unclear if it was ablating the sample or ablating the resin. So I think that using mostly recruits is easier, it's easier to prepare the samples, it's less time consuming, and you get more reliable information from it, which is why I looked at both larvae and recruits. And my other assessment is that this type of study is really important to do at first, but does not need to be repeated constantly. I think once we have this information, we can really just rely more on hydrodynamic models.
And then you're saying about, in terms of ecological effects, they could be vector for pathogens. Is there any pathogen for this species?
Yes, there is. It's M. perfringens, and it actually has the potential to go to aquaculture systems. This is a big problem. This pathogen is a big problem for these aquaculture rafts because if the oysters and mussels get infected with this pathogen, then they aren't going to be as good for human consumption, and this species is a vector for that pathogen.
And lastly, you're talking about the management actions, so it would be physical remover. Is it removing them?
Yes, so a lot of the study sites are accessible by foot because this species does prefer to be very close to the river mouth.
Okay, and what would be the end of, if you remove them, what are you doing with them? Is there any end for that?
That is a very important question, I think. These mussels are not eaten by people because they're so small, which is another reason why they are such a prominent invader. They don't really have any, they have one natural predator when they're in their recruit form, and that's a crab. But the crab shows a preference for the native mussel. So maybe this is a great avenue for future research about what we could do with those shells.
Okay, thank you.
Yeah, so thank you for such a nice piece of work. A lot of information to process. I didn't have the opportunity to read your manuscript, but I think it's really interesting topic and the way that you approach it. You explained really well, so congrats for that.
Thank you.
I have just a few questions. It's known, for instance, compare for the same locality other sessile invertebrates with the same techniques to see how they perform. I mean, what I'm worried is like which might be the overall heterogeneity of your system.
That is a really great question, and that is why I think preparing for future climate scenarios and understanding the climate models is really important for management. I think the main finding here is that they are dispersing between RIAs, and so now that that has been established and that it is likely the upwelling that's enabling them to move out of the RIA, we can kind of monitor what's happening with the upwelling index on a yearly basis, as well as precipitation, which is another factor which impacts water movement both within and between RIAs. And this can then be synthesized with the results from this study to predict if there might be more self-recruitment, like if there's more precipitation or reduced upwelling conditions, or more movement between populations and RIAs. And if that were to happen, like if there is a year with higher upwelling, or if there's a year with very low precipitation, then I think that really focusing monitoring efforts on the northward RIAs would be ideal. And then if you're observing that the species is continuing to expand, and if there's funding for it, then deploying some active management techniques.
Okay. And as you know, Ronnie, this was a huge problem. These struggles for northern Spain. So even in the studios we have some, but in the last year, what we were seeing in some of the rain, for example, in Navidad, that they are declining in population.
Oh, really?
So, are you planning to expand these, or would you like to expand these in more? Because in different situations, could be telling you different story about this invasive.
I think expanding monitoring is really important, and then having monitoring just as the first step for planning for management, and focusing those monitoring in the northern RIAs.
And then, do you know if there is some genetics on that? Because other line of evidence suggesting this sort of movement or interaction of recruitment in that part of the distribution.
For this species, there hasn't been this type of study yet. The species has really only been documented, and the population density has been mapped individually, like in different studies between these two RIAs, but there has not been any connectivity studies about this species in these specific RIAs.
return. So congrats for them.
Yeah.
We have time for some questions, 5 minutes. So if any, no? Do you have any questions? Does the jury have any questions that they wanted to?
Okay.
Dylan has a question.
Okay, if there would be something that you would change from your older thesis, what would be and why?
Thank you for that question. Okay, well, I would start learning Python earlier because I did that. I did that when I was trying to process my data, and I never used it before, and it took a really long time to learn. So I was working a lot just to figure out how to process my data. And then from a methodological perspective, some studies coming out of the east coast of the US about the eastern oyster have mounted samples to double-sided tape rather than I used a resin epoxy to mount my samples to the slides. I did try it with a tape, and it was, but toward the end of my lab work period, and it was easier. So I think in the future, maybe focusing some studies on if there's a difference in the quality of the signal region if you use resin epoxy versus tape, and then if there's no difference, then really mostly using the tape moving forward.
Thank you so much.
Anyone else? No? Then I have a question. If you would have fundings for your next research, what do you think would be the first thing that you would want to research in this topic?
About this,
Yeah.
I
You
would,
have some future research, but what is the most important thing, the next step?
I think having a greater understanding of larvae behavior, because that has not been studied for this species, at least that I've found, definitely not in the RIAs. For example, how different climate conditions or extreme weather events might impact larvae behavior, how larvae move through the water column, if they have a preference for the surface or the deep water, because that all impacts how they circulate around between RIAs, and then also just what conditions they're able to survive.
Okay, if there are no more questions, so my computer died, so I don't know if the live chat has questions. Do you have it in live or? Then we can continue with the next presentation. Yeah. Woo! From Bible, we move to stations with the presentation by
This. Yeah, okay, so hello everyone, my name is Ricardo and I will be speaking about my thesis on species distribution modeling of cetaceans in Cape Verdean waters. So Cape Verde is a region that is important for various cetacean species. The humpback whale, for example, it is the only known breeding ground for the species that receives both northern and southern populations. It also holds really large populations of various odontoceti whale species, many of which are likely resident species. But we have very little information on the cetaceans in this region, and we don't know much about their ecology and their distribution. So this is why we can also use, for example, species distribution models to fill in information gaps. So species distribution models use presence and absence data combined with environmental variables to predict a species distribution to a study area. These distribution predictions can either be what we call probability of occurrence if you use both presence and absence data, or if you only have presence-only data, you get distribution predictions on habitat suitability or habitat preferences. You can predict them using statistical algorithms. You can either use one algorithm or you can use a mix of various algorithms in the model, which is what you call the ensemble model approach. And it is the approach I took. So I tried to develop the first species distribution models for cetaceans in Cape Verdean waters, and I used both presence, absence, and presence only data in an ensemble approach to try and get a complementary analysis with both types of data. And this way I try to get new insights on the species ecology that we can use for research and conservation in the future. So my study area involves all of the Cape Verdean exclusive economic zone. plus some adjacent areas in the high seas and other African EEZs around. And this way I could use all the national waters while at the same time I covered a broad range of environmental variables. Then I tried to get as much data as possible. I contacted a multitude of sources. 13 of them share their data. It includes systematic data collected in citation service and opportunistic data coming from citizen sciences and whale watching, fisheries, cruise ships, and so on. And I also tried to use a large sample extent of 25 years, so from the year 2000 to 2024. Then when I processed and standardized all my data, I made 3 modeling data sets. One was the presence data set for the presence only model that involves all the sightings, all the occurrences I had. The second was also presence data set, but for my presence absence models that only had scientifically collected data, and absence data also for the presence absence model that was computed using the effort logs from the scientific service. Then, in my data set, I picked the 5 most frequently recorded species as my study subjects. They were the humpback whale, rough-toothed dolphin, the short-fin pilot whale, pan-tropical spotted dolphin, and the bottlenose dolphin. And for the environmental variables, I tried to use as much as possible that were recommended by literature. I chose 13, but there is also the problem of spatial correlation. So to test for that and multicollinearity, I used the variable inflation factor, a VIF analysis to find multicollinearity among variables. And I had to take out 4 variables from the model because they had a lot of correlation. And my ensemble models, once again, I did 2 types of ensemble models. One was with presence absence data, and I use commonly used algorithms, GAM, RF, and Mars. And for these, as I have both presence and absence data points, I could get the probability of occurrence estimates for a species throughout the study area. For the other model, I only had presence data, and I use the same modeling algorithms plus MaxM, which is very popularly used, but only available for presence-only models. So presence-only models, they don't give absence information. We don't know where the species is absent, so we can't get probability of occurrence. Instead of that, we get outputs on habitat suitability or preferences. And to test if the models made ecological sense, if they were trustful, I used 5 metrics to test them. The AUC was the most commonly used one. It is the threshold independent metric, but sometimes it can be a bit misleading in the results, so I also backed it up with 4 other threshold dependent metrics that were the omission, sensitivity, and specificity rate, as well as the Cohen's kappa coefficient. Now for the results, we managed to get the largest data set on citation occurrence ever made for Cape Verde, almost 1,600 sightings compiled. About 80% of those sightings belong to only the first 5 study species. Humpback whales were by far the most recorded species, about half of the records. The other ones have very small occurrence numbers. And here you have data originating from scientific service, and you can see that it's a very small portion of the total data set because there's very few scientific studies on cetaceans in Cape Verde. Now, before we speak of the model outputs themselves, I just wanted to take into consideration a couple biases and limitations on the data. First of all, as they came from multiple sources, they have multiple methodologies. So some of them were collected in research vessels, others came from artisanal 5-meter wooden boats. We also have systematic data. A lot of them probably had false predictions, so false absences because all the data were collected visually, and if cetaceans were diving, they were probably not spotted. And also there were likely spatial bias associated because a lot of my data came from citizen science. So we have to analyze the next models a bit cautiously. And for the presence absence predictions, you will notice they're very small hotspots. So they're not exact predictions, but they're instead, we use them to identify hotspots of cetacean occurrence. But generally, the models performed really well with all the metrics. All the algorithms except GAMS had good performance. Except MaxM. MaxM had a bit of a weak performance, but still, it didn't affect the final ensemble models. All the ensemble models performed well. And the presence absent ones had slightly lower performance because they had a much lower amount of data available. Starting with the first model is the presence only model for the humpback whales. So we had high habitat suitability on shallow waters on the eastern islands and also in this offshore area, which is the John Valente Bank, the largest coral reef in Cape Verde, but it has a small amount of model uncertainty when predicting it, so not all the algorithms agreed on those predictions. we notice that the main variables influencing their distribution is shallow waters, shallow bathymetry, proximity to the coast, and slow water speed. This is usually what humpback whales are looking for when identifying their breeding grounds. And for the occurrence probability, as you can see, the hotspots are way smaller because of the small data, but you can notice that this area west of this island, Boa Vista, is very important for the species. And the João Valente Bank, the coral reef, also has good probability of occurrences above 0.5 or 50%. Uncertainty in this model was somewhat a little bit, but mostly in offshore waters, far away from the main spots. Then we have the next species, the rough-toothed dolphin. Also had a lot of preference for the eastern islands, but very close to the shore, so very coastal distribution. It generally had somewhat of uncertainty, but it was not in the main prediction hotspots. This very coastal distribution is usually seen in rough-toothed dolphins in other volcanic islands, like Madeira too, or Hawaii. They like to be close to the shore. And we have somewhat the same predictions for the probability of occurrence. So they also really like this coastal area west of Boa Vista, and that's pretty much it. So the fact that we have a very small territory where they congregate, it means that most likely we have a resident population with strong site fidelity, but once again, there was never a study on this, so we can't confirm it. Then the short-fin pilot whale was the one with the most offshore distribution, so they really liked waters over inshore shelves and with big gradient seam depth. So they were a lot concentrated in slope areas and in islands where the bathymetry increased very fast. And you will also see the same thing reflected in the next model, but I do have to say that this was one of my weakest models because I had a very small sample size, only about 20 or 30 occurrences. So it gave a very big probability of occurrence, but somewhat big on this offshore area, but it also had a lot of uncertainty involved. So we can't actually take this with confidence. We should just instead look at these very small patches, very close to the Santiago Island, which has a really steep bathymetry, and at very low uncertainty. So we can identify a really important habitat for the species west of Santiago Island. Then we have the pantropical spotted dolphin that are a very coastal distribution. You will notice that suitability was very high close to the shore and decreased with distance and depth. And uncertainty also increased the furthest away from the shore. So this species is also usually taken as a very coastal species in other regions like the Antille Islands or Hawaii. So it also goes in line with previous studies in other parts of the world. Once again, in this model, the probability of occurrence, we had small sample sizes, so distribution were a bit harder to take seriously. For example, we had significant probability of occurrence offshore, but they had a small amount of uncertainty involved. And it's a bit small to see in the map, but also around a lot of the southern islands we have some really high hotspots of occurrence, which had really low uncertainty, so we managed to identify important hotspots here that can be trusted. For the last species is the bottlenose dolphin. So the bottlenose dolphin also had a really strong coastal preference for habitat suitability, really high habitat suitability value in coastal areas. And we also had some somewhat strong values, above 50% in these offshore areas. We had very low uncertainty, only at 10% or 0.12. So the models generally had good results. A lot of agreement between different algorithms. And for the probability of occurrence map, also it gave some probability of occurrence offshore and identified some really good hotspots in some of the islands. So São Nicolau, Santiago, Fogo, and Maio Islands are all reported to have good amounts of bottlenose dolphins, and the model show it. So we do see that we have a lot of distribution both close to the shore and further away. And if you look at the occurrence points which are in the paper, you will notice that we have occurrence offshore in here. So we might either have two bottlenose dolphin populations, one offshore and another coastal, or just one population that travels a lot throughout the area. And to speak a little bit of the implications that you get with the models. You might have noticed that pretty much all the species had a lot of affinity for coastal waters. So it shows that the coastal waters around Cape Verde, just like every other oceanic islands around the world, like Hawaii, Madeira, Azores, make up really good habitat for cetacean species. And the models also identified the new hotspots, for example, Santiago Island that we were speaking, João Valente Bank. All of those regions have very low cetacean surface, but the models still identify them as important spots. So you might have noticed we need a lot more data in this region, so we will need to carry out more surveys, and the surveys they should try to validate the hotspots we find. So to try and see if the hotspots actually make sense. If they are validated, we can use them to identify important areas for conservation management. We can, for example, create marine protected areas or important marine mammal areas with those validated hotspots. And it would also give further insight into the species ecology if we did more research on those hotspots. Also, this study did not take temporal variability into account. So I didn't take, for example, changes in environmental conditions seasonally, and future studies should try to investigate that to see how difference in seasons or climate changes through the year will influence the distribution of the various species. And now to conclude, this was the first species distribution model study done in Cape Verde, and although there were a lot of data limitations, they gave like preliminary results and a lot of new insights on the distribution of cetacean in this data poor region. And we should, of course, continue trying to get more data, but anyway, the results do support the importance of oceanic islands like Cape Verde to various other cetacean species. Using both presence absence and presence only data is not a very common approach. Usually you should pick one or another, but this study showed that when you have a very small amount of data, using both of them can actually be beneficial if used complementary. And of course, future studies we should try to test for temporal variability. So we should try to see how the distribution changes throughout the seasons, and this way we can get more accurate distribution predictions. And yeah, that's it. Thank you very much.
Thank you, Ricardo.
Thank you.
Really nice presentation, and as I understand, it's kind of pioneering study in that area. I have only one measure question.
Yeah.
Do you think that the distributions you observed could be also related to anthropogenic impacts or activities?
Yeah, for sure.
Could you match?
It's like so we had spatial bias in this model because as a lot of them came from whale watching areas, fishery zones, areas near the coast. It does cause a certain bias in the model. That's why we would need more data collected through scientific surveys that try to match each area equally. But I didn't include it in this presentation, but I also had the data collected through systematic surveys. Most of the surveys covered offshore areas, far from the coast, and still about 80% of the sightings happened in coastal areas, so less than 12 nautical miles from the shore. So even though we had a lot of offshore coverage, most sightings were near shore, which support the results from the model that near shore areas, coastal areas, are likely the most important for the species.
For example, the coastal areas, in coastal areas, you could find relationships between the distribution of dolphins and fisheries. We observe this kind of relationships just in front of here on the Adriatic Sea, in front of Ancona. So I think that this could deserve attention for your future research.
A lot of SDM studies with cetaceans, they try to investigate what is the relation between fishery areas and cetacean distribution.
Or touristic activities, especially just to give an example.
I wanted to do that, but also they usually use data available online on fishery distribution. For Cape Verde and West Africa in general, there's not that much data because a lot of the fishermen use wooden boats without tracking devices, so you don't really know where they usually are. So it was a bit harder to test the effect of fisheries on the distribution of the areas and local circulation.
Thank you.
Okay.
Hi, Ricardo. Thanks for your presentation.
Thank you.
I was just wondering, why did you not take seasonality into account? Did you not have this kind of metadata from your data?
Yeah, I wanted to take, but the problem is that we have very small sample sizes at the moment, and they're mostly collected in the summer because Cape Verdean seas, they tend to be quite rough, and the vessels they usually have available are like either small fishing boats or sailing boats. It's not big research vessels. So if I try to test for seasonal variabilities, I'd have really small samples for every other season but the summer. But that's why if we continue collecting more data, and especially if we try to collect it in other seasons, we might have enough data to check the seasonal variabilities.
And then, so what's the time span of your data? What was you had probably for multiple years, so how old is your data and or how young is your data?
25 years, from 2000 to 2024.
Okay. And did you not feel you could have done some of that collection as well, like while you were doing your thesis? Was there not no opportunity for you to?
You mean like to collect data myself?
Yeah.
Initially, that was the thesis plan, that we would do some service to collect it, but we didn't get financing for a boat. It was a boat supposed to come from Spain. It didn't come after all. So I just had to collect data from as many sources as I could. But usually, for you to have an idea, citation surveys usually happen in Cape Verde every 5 years. So it's not very often that you will have the availability to collect data on a research vessel. So you have to collect data from already existing studies.
And you said that there's systematic surveys done. Who does those?
So one of them is the Irish Whale and Dolphin Group. For example, I had this photo. For some reason, they like doing citation surveys back in Cape Verde. This is one of the photos they took back in 2019. There were other surveys done by, for example, the Elasmobranch organization. It's mostly foreign research organizations. So Elasmobranch from Spain, and Thesis, which is a Portuguese project. But yeah, it's mostly foreign research projects that every now and then get enough financing and they go and try to collect some preliminary data in Cape Verde.
And lastly, how long did it take you to analyze your data?
Well, let's say maybe about 2 months. I started initially analyzing it, but the thing is that not all of the sources provided the data at the same time. For example, some of them provided them after less than a week after I asked. Other sources took a month to send me the data. So I already started analyzing the data, let's say in March, February, but I only had enough data to do a serious analysis by the last 2 months of the thesis, something like that.
Okay. And what does the future look like for you now? Do you want to continue on this path?
For now, for this study, it's concluded, it's done, so we can see if we can make it public. But in the future, it would be really nice to have more data. For example, this year they're going to have the largest citation survey in Cape Verde. It's going to be the same boats from Elasmobranch. It's going to be doing a research for a month there. So it should provide a lot of data. And when we have enough, probably tests for the same species again and get better outputs. And for other species like the sperm whale, it's quite common in Cape Verde, but you need to use acoustic methods to detect it. So if the next survey is actually use hydrophones, we can have, for example, acoustic data to model sperm whale distribution or big whale distributions too. So that would be a way to look for the future.
Thank you.
Okay, Ricardo. So thanks for your presentation. Really nice work. I have some questions related with the data. What kind of filter did you use? I mean, because you have different sources.
Yeah.
So which filter did you use to trust the data that you're working with? If you can comment on that.
Yeah, so let me go it. For the data I collected, even though they were a lot of them came from citizen science, they were validated by scientific project. Even the fisheries data, for example, the people involved with it, they received training by local environmental NGOs on how to collect citation data. So they already used certain methodology and some information. And I spoke with the NGOs that were working with them, and by their own words, they were valuable data. They were using the data collected through fisheries. Then for other data, for example, cruise ships, they usually, I'm speaking most of like expedition cruise ships, so people looking out, going for nature research. They always have nature expeditions. So they always have a research on board. So the data coming from here actually came from biologists on board. Same from cargo ships and data repositories like I used iNaturalist. Data on iNaturalist before it is available for scientific use. It needs to be approved by various specialists. So all of the data had some sort of scientific validation.
But did you apply some filter, or did you find it necessary to apply certain filters?
For some, like for the fisheries data, a lot of them had the GPS was quite off. For example, when I plotted it, had data appearing in the middle of the way far offshore for small fishing boats. So I spoke with a couple fisheries technicians and local biologists on what would be the maximum distance they get from shore. And they gave a conservative value of 4 nautical miles. So the small boats don't go far than 4 nautical miles. And we put a buffer of 4 nautical miles for the fisheries data. And every fisheries data that was away that, further than 4 nautical miles from the shore, I removed it from the analysis.
The other thing is that I'm thinking, did you have the opportunity to test the weight of each of the sources in your model? So how it's contributing, maybe some bias, not bias, but different weights of different sources to see how that is feeding your models?
Yeah,
Actually test that?
I did that mostly in visual analysis, so mostly looking at the data and on Excel files, for example. And for example, some of the NGOs, the ones that are more involved with citation research, they provided more than half of my data set. The thing is that they are only concentrated in 2 islands, those NGOs, so Sal and Boavista Island. So they had a really big weight of data coming from those 2 islands. And then other, for example, cruise ships provided very few data. We had about 30 or 40 samples coming from them, while from NGOs they had almost 1,000 samples coming from them.
It's really biased.
Yeah.
And the other thing, do you have, I mean, normally when I'm doing some modeling, so I like to have some sort of internal controls, yeah, to know that there's some things that you, I mean, if you're working with certain species, you need to pick up that.
Okay.
Do you have those sort of controls, internal controls, that you know that there's some pattern that should be definitely there? At least for some of the species that you're working with?
Not sure if I understood. You mean like to identify patterns that should make sense?
Exactly.
Okay.
Yeah.
So mostly doing literature research. So every time I got the model outputs, I would check previous SDM studies or other kind of distribution studies to see if they match. And for all the species they match. So the humpback whales everywhere in their breeding grounds, they really like shallow coastal waters. This is what we saw here. The rough-toothed dolphin, it was a bit more tricky because near islands regions, they really like coastal waters, but continental settings like Brazil or Mauritania, they prefer offshore waters because the shallow depth is much more extent. So with these species, it kind of depends on the region, so it was a bit harder to validate the distribution. Pilot whales, they really like slope areas, so the areas you see here are the slope of the island's shelf, and it was what the models predicted. Another species, the pantropical, some regions of the world they like coastal waters, other regions they really like offshore waters. So it's matched with the regions of the world where they like coastal waters, but it didn't match with the regions where they like offshore waters.
Okay, thank you very much. Great job.
Thank you.
Hi, Carla, thanks for your great presentation. I was wondering, well, I was thinking about a way for increasing your observations. So explore the use of remote sensing data, and you mentioned that use of acoustic data would be a way to be able to detect them. But I was wondering if you know of any studies that have used satellite data or drone data to monitor situations, and if this could be used in this type of modeling, and if so, do you know if there's any uncertainty that could potentially improve the bias in your different sources of observations?
Yeah. So if I understand you correctly, there's not data collection using, for example, satellite data on cetaceans for Cape Verde at least. There's also a lot of times you can do like airplane census, airplane service, which has also never happened in Cape Verde. So the data is usually either collected from land, from the islands, or from boats themselves. Also for the humpback whale, for example, one of the hotspots it gave was this island, Sal, which is not usually talked about for humpback whales, but there is a paper that was investigating their distribution through interviews. They interviewed a lot of airplane pilots landing on Sal Islands, and they would say that they would see a lot of humpback whales near the island when they would land. So that's the closest to an aerial survey I could get.
One more small question.
I'm not sure if you know that, but I was just wondering which species is like the most common one, and if these species are like resident to the paper, or if they also like go to other, for example, in winter.
So like abundance-wise, it is not known. I had the most common species, recorded species was the humpback whale, but it's a very small population, only 300 whales, while the dolphins you have thousands of them. But the humpback whale gets a lot of study focus in Cape Verde, so we know the population for them. All the other species, we have no idea what's the population, the abundance we have in Cape Verde. Okay.
Did you have a question or?
Oh, I was just gonna ask if we have time. I don't really know that much about whales. Why do some of them prefer like the inner coastal area and some prefer like the outer shelf area?
It has a lot to do with the way the environment influences them. So the humpback whale really likes coastal areas because we have slow water speeds, it's shallow, and it's really good place for them to raise their calves, to give birth, and for the reproductive season. The short-fin pilot whale really likes deeper areas because it's where you will usually find their prey, which are deep diving squids and deep diving fish. So that's why they will prefer deeper areas. And then we have dolphin species like bottlenose and rough-toothed dolphins. They really like coastal areas too, for feeding, not breeding like the humpback whale. But it's mostly because they feed on coastal species, like coral reef species, for example. So they have this tendency to prefer coastal areas.
Thank you.
You're welcome.
Thank you very much. As well, and at the end of this session, we'll have a coffee break, and then we come back to the session.
All right, good morning. I hear you are online. So we are ready to start. We will present myself. Maybe there are some people in the audience that don't know me, but we are streaming online. So it's a great pleasure for me to be the chair of this session. I wish I could be at all sessions. I'm Luana Monteiro. I'm the main coordinator of the IMERC Master. Before I introduce the speakers of this session, I would like to present our jury and to remind that we have 15 minutes of presentation for each student, followed by 15 minutes of questions, discussions with the jury. If we have time, then I will give the chance for the public first present here to ask questions, and then we will assess if there are questions from those following us online. So my colleagues from the jury here today, so my colleague from Oviedo University, professor and local coordinator of partner Oviedo University, Gonzalo Machado Scafino, try my best with the names. My colleague from the University of Western Brittany, UPO, Nicholas Jerry, and my colleague Sylvia Bianchelli from our local host University Polytechnic Denmark. And it's my great pleasure to invite our first speaker for today, Joanne Waters. Joanne is going to tell us about population genetics of the undulate ray Raja undulata in the South Bay of Biscay, Northeast Atlantic. And Joanne, for you and for all the speakers, I will stand up and then you have 3 minutes left, and I will make you a sign. You try to make it visible if there is 1 minute left, just to guide you. And success, the floor is yours.
Hello everyone, I'm going to present the results of my master thesis that is titled Population Genetics of the Undulate Raja undulata in the South Bay of Biscay. And starting with a little introduction about Elasmobranch, so they are cartilaginous fish, they are top predators and keystone species. They have a slow life history, meaning that they have late maturity, low fecundity, and long lifespan, and that makes them highly vulnerable to overfishing and habitat loss. The decline that we're seeing right now in the Elasmobranch population is mainly linked due to the increase in fishing pressure, both targeted and as bycatch. And there's a real complexity in the conservation of Elasmobranch due to their species-specific ecological traits, to regional management, but also to socioeconomic factors. And I would like to insist on the fact that there's less than 5% of Elasmobranch species that have been assessed using direct or indirect fishery stock assessment methods, and this is very low. This study, of course, focused on only one Elasmobranch species, that is Raja undulata, the undulate skate. It's a coastal species that is found in the Northeast Atlantic and the Mediterranean Sea, and is usually caught as bycatch in commercial trout fisheries. It also exhibits strong site fidelity and localized aggregation. And I want to mention that it was previously classified as endangered on the IUCN Red List, but now after reclassification it is classified as near threatened, close to vulnerable. And the interesting part is that this reclassification was made on the bare observation of an increase in landings and not on any stock assessment, for example, using genetic data. So when we look at the distribution of Raja undulata, we need to understand that there is a multi-scale regulatory framework. For example, when we start at the European level, it is managed under the Common Fishery Policy, and they take advice from the ICS. that is now saying, don't target Radio Undulata in the South of Biscay because we don't have data on stock status and on the resilience. And as I said, it's mentioned on the IUCN database. But now when we look at the Spanish level, we don't have any mention of Radio Undulata on any list. We don't have it on the list of species under special protection regime. And it's managed using quotas, meaning that they open and close the fishery depending on the utilization of those quotas. And now when we look at the regional level, for example in Asturias, we don't have any mention of Radio Undulata on any list, but actually there is no mention of any marine species. That's a bit questionable, and it really depends on the region in Spain. And so when we have like all those inconsistencies, of course it doesn't make it clear to know what we actually have to do to manage the species. As just a little reminder of what we consider as standards for sustainable fisheries, here I'm going to focus on the Marine Stewardship Council, but they have three main principles. And the first one, very easy, sustainability of the stock. And when we look actually at what they say for sustainability of the stock, they mention at different moments the genetic structure of the stock or the genetic structure of the landings. And they also talk about population, connectivity, differentiation between the population structure, and all this necessitates genetic data. And the gap here is that in the survey of Biscay, we don't have genetic data about Radio Undulata. We don't have information on population structure and connectivity, and this limits the establishment of science-based and efficient management plans. So the objective of this research were actually to know whether the population in the survey of Biscay is genetically distinct or part of a broader connected population. All of this to inform adaptive management strategies and, on the long run, ensure the sustainability of this valuable marine resource. How did we do to answer those questions? Actually, we use a dual approach, meaning that we use, on one side, mitochondrial co-gene barcoding, and on the other side, microsatellites marker to assess connectivity and diversity of Radio Undulata in the survey of Biscay. Moving on to material and methods, we worked with 100 individuals. 64 were collected in Asturias, and 36 were collected in Galicia by the Spanish Institute of Oceanography. And I want to insist that this sampling was made in collaboration with fishermen and market workers. Here, it's some pictures of the sampling I took part in, that was in Asturias at La Nueva Playa. And for the lab workflow, so we started with sample collection, the preservation, the extraction, quantification, and purity to have some thresholds. And then we had two parallel protocols that were going on, depending on the markers we were working with. So, for mitochondrial DNA, we started with normal PCR to amplify co-gene, we had gel electrophoresis to check for successful amplification, and then everything was sent for sequencing. And for the microsatellite analysis, we used multiplex PCR, meaning that we had sets of primers together. We also checked for successful amplification, and we set everything for genotyping. Then we had different bioinformatics pipelines that I'm not going to develop in details here, and I'm going to move directly through results and discussion. So, starting with mitochondrial DNA. Mitochondrial DNA is transmitted via the mother. It gives insights into historical connectivity and the dispersal of females. And here, in our data, we actually detected only 2 haplotypes. So we had very low haplotype diversity and also very low nucleotide diversity. Even when we look compared to other species, Radio Undulata is the extreme corner. This low mitochondrial diversity can actually indicate long-term stability or recent bottleneck. But we checked for bottleneck using Tarima and full neutrality test, and they were not significant, showing no sign of recent expansion or contraction in our population. Then, when constructing the haplotype network to have a visual representation, we have those 2 haplotypes that have an unequal distribution across localities, and we have this significant PSC value that suggests genetic structure, genetic differentiation between our sample location, and highlighting the unequal distribution of maternal lineage. When we saw this low number of haplotypes in our sample size, we decided to construct an unweighted haplotype network based on the data that we could find online, so on GenBank. And we actually realized that there's a low number of haplotypes that is described for the species overall. And when we take all of this information together, we can actually think that it's more stable or historical processes that are actually responsible for the soil diversity. But we have genetic structure, we can see it, so we need to take into consideration this genetic structuring of the maternal lineage for conservation and management efforts. Then when we look at microsatellites analysis, so just a recap. Microsatellites are a nuclear marker. They are inherited from both parents and they give more information on recent and ongoing connectivity. We started with 12 different loci. 8 were described for Raja undulata and 4 were described for Raja clavata. But they were already tested in other studies for cross-species amplification and well working, so we decided to keep using them. And then we had different steps where we had to be careful about how our markers were behaving. And we realized that we had to delete 3 of them for non-specific peaks or problem of amplification or even technical inconsistency that were reducing the reliability of our marker. So the analysis were performed with 9 different loci. In terms of results for our microsatellite analysis, in general, we saw average diversity and moderate difference when we looked at observed and expected heterozygosity. And then when we looked at the FIS value, so the inbreeding coefficient, we had significant positive inbreeding coefficient, both in the global dataset, but also when we looked per location. And this underlines the possibility of having localized inbreeding, well effect, small effective population size, or local demographic fluctuation. And then we looked at the FST values, the fixation index, that was low but significant. So it shows the level of structure. And it was low but significant when we use FSTAT, but when we use amobinarity, it was similarly low but not significant. So we have to reject the mixture because we got a signal of structure. But this is probably a slight genetic structure that may exist and that actually does not reach statistical significance when we use different methods. We also did check for bottleneck in our microsatellite markers, and we did not see evidence of recent population bottleneck. So this highlights the fact that even if population have gene flow, there can still be internal processes that are affecting their evolutionary resilience. Then we did some visual analysis, and we started with structure analysis that is using the Evanodotaki methods that is supposed to give us the most probable number of clusters in our data. And the answer was 3. Okay, so then we checked the structure plot, and when we looked at those three supposed clusters, we don't see clear genetic structure. It's like high mixture. It's a bit contradicting, but we also have to keep in mind that the Evanodotaki methods doesn't test the hypothesis of having only one cluster. So we cannot have the detection of artificial structure. But then we looked at PCA. And same PCA here, we don't see a clear genetic cluster, we have like high overlap, so it doesn't seem to highlight having genetic differentiation. But when we did the DAPC, that maximized between group variation, we actually see that it was supporting the hypothesis of having 2 or 3 clusters. But same as the Evanodotaki methods, it doesn't test the hypothesis of having only 1 cluster. So, you might get a bit lost and think, but is there a structure? Is there no structure in the population in the South Bay of Biscay? This is not very clear. I get it. It was not very clear for me too at the start. But actually, what we know is that we got the signal of structure. We had analysis that said there is structure. We had the FST value, we had DPC, the Evanodotaki, and even though we have some analyses that are not giving us a clear signal of structure, we cannot just say, no, it's not working in all of them, so there is no structure. No, we have a signal that is probably not strong enough to be detected in our analysis. Okay, now we look at mitochondrial, we look at microsatellites. What happened when we look at it together? Actually, we have a stronger differentiation when we look at mitochondrial marker than when we look at microsatellites marker. And this made me think about the male-based dispersal, when you have a stronger distribution, larger distribution of male than of females, where females tends to be staying at the same place. And this actually has already been observed in Elasmobranch, mainly in sharks, but sharks are cool, so we study them more, but also hypothesized in rays. And one of the rays is the Thornback ray. They got similar data as what I have here. And they hypothesized that maybe there is male-based dispersal. So the question is, do we have male-based dispersal for Raja undulata? It's a nice hypothesis, but we have to be careful. Because the difference that we're seeing here in the level of differentiation may be due to intrinsic difference between markers, the way they're inherited, also the power of the loci we decided to use. And in this study specifically, we have a limited scope of the sampling design. We have to delete some markers, so it's relatively low number. We have no other analysis that could confirm what we're seeing. And it's also an understudied species. So we have a lack of information about connectivity and historical demography that could help us understand what is actually happening. This underlines the importance of having a multidisciplinary approach to understand what are the processes that are shaping the structure that we can see, and what are the implications for conservation. As conclusion, I would like you to remember that we got, in terms of maximum data, low diversity, but singular structure. For nuclear data, we got average diversity, and we did got a singular structure, even though it was not everywhere. This suggests partial connectivity between Asturias and Galicia. In terms of conservation status and reality, in this study we don't have evidence of connection, and this can actually question the species resilience. It can also question the downlisting by the IUCN to near threatened, because actually we may have local vulnerability that we're underestimating. And also, even though in Spain we have those quotas that are supposed to manage the fishery, with the project I'm working in, so the Shark Crema project, they did detect mislabeling. That is actually quite high, and even higher when we look at Raja Undulata. And this may bias landing statistics on which they're regulating the fisheries. This may lead to some question about the effectiveness of those measures. What has to be done now? What do we need to do in terms of research and management priorities? First, we need to keep investigating the local and regional fine-scale structuring that we're observing here. We need to explore seasonal movement and also this potential sex-based dispersal. We need to use other methods, such as telemetry, coalescence model, expanded genetic sampling, because here was quite reduced, but also maybe having information about the sex of the individuals. We need to also improve landing traceability and update protection framework, nationally and regionally. We need to have something that is coherent from Europe to regional, Europe to Asturias. And we need to incorporate genetic data into stock assessment and policy, because we saw just before that to have sustainable fishery, we need to have information about the stock status, and we cannot have clear information about the stock status if we don't have genetic data. So this work is actually just opening doors. It's not answering questions. It's opening doors to the fact that we have to keep searching and we have to keep working on this to reach sustainable fisheries and to be able to manage correctly those populations of Elasmobranch that are near threatened or endangered, but there's something happening clearly. I would like to thank my supervisors, Laura, and everyone that makes this project possible, everyone in the team of the Shark Map. And those are the references for my presentation. You have more in my manuscript, of course. Thank you for your attention.
Thank you very much, Joan. Keeping nicely on time, but also for a very enthusiastic presentation and about such a relevant topic, but challenging as well. Now is the time to give the word to the jury. Do we have a volunteer to start asking questions? If not,
me? Okay. To start with, I'm not at all a population geneticist, so disclaimer, but yeah, I still have a few questions. And I guess I'm more of a stats guy, so you know, something I'm interested in that aspect. So you found one of the results you had was low diversity for mitochondrial DNA, right? And I was wondering how and if that could be related to your sample size, because you have 100 individuals.
Yeah, so actually when we look at the haplotype network that is here, so I have, it might seem that I have a low sample size, but normally for population studies it's already enough to be considered like interesting. Important. The thing that I have is that I have unbalanced sample size. I have 64 in one set and 36 in the other, and I was getting a bit worried, but actually when we see, it would have been more worrying if I had the opposite result. So here I have this rare haplotypes that is actually mainly present in Galicia, where I have the lower number of samples. So if I had the other statistics, I would be getting more worried about my unbalanced sample size, but here I don't think it impacted that much the reliability of the data I'm seeing here. Also because when we compare in general, there's like not a lot of diversity in terms of haplotypes. So.
Okay, I have another question. So you've got this. So you've got this like sign of structure, but not necessarily super clear on a bit of the macro. And one of the way you approach it is by basically clustering analysis and trying to get clusters. And I was wondering, sometimes you have structure, but it's not really clustered. It could be like more continuous or more gradients. And have you explored that, or do you have any ideas?
So we kind of tried to explore everything because I was getting a bit lost, as I said, about having structure, having no structure. So we kind of tried a lot of different things to see if what we were observing was actually what's going on. Because here we didn't find any we didn't explore more like the biological relevance of the structure here because it doesn't seem that it was, at least in microsatellites, based on the sampling location. But we did see that, for example, one of our hypotheses was that here we have a clear structure in mitochondrial. So we were thinking that maybe if we actually sort the data in another way and we start exploring based on the mitochondrial structure here, so we can split the data in that way. We also tried to remove some markers that maybe had were too extreme in terms of results, and we didn't see any clear way of interpreting this right now, so we actually just want to expand the sampling to have more information and to see it more globally, because as there's not a lot of other studies, we cannot compare what we see here in other analysis.
Okay, and I guess so related to that, I was wondering in terms of ecological interpretations. So you got this some structure signal, but it's not really special, or at least not at the scale of Galicia. And how do you interpret that?
We know that there are still more things that can be tested to actually. I'm not an expert in oceanography of Asturias, but I know that there is at some point a cabo or something that is. There are also some studies that there could be structure related to that. We also know that the oceanography physics in Galicia can also impact the population structures. So for now, I don't have conclusion about why, but we're thinking, and we're also. I would like, if I could, I would like to have information about the sex. Also, maybe have information if we have the sex-based dispersal, looking at maybe if this is leading the differentiation. I think there's a lot of things that we could test because right now we don't have clear reasons to say, okay, this is not following the sampling location, but are they even distant enough? Yeah. And so now there's further studies that are going to be made in the Shark Remap project, also with genomics, less of SNPs, to also maybe see what is going on, because my markers also are maybe not the most efficient to try this. And to answer this question, having also more sampling from Portugal, from the UK, to have a bigger picture and maybe try to understand the biological meaning behind that.
Thank you. Sylvia? Thank you for your super nice and enthusiastic presentation. You did a really good job, to my opinion. I have a couple of questions. The first one is, in your opinion, which could be among the doors that you opened and the questions that you opened for the future, which could be your priority for the next study?
I think for the, I'm sorry, I'm going to faint now.
We understand you. We are on the same boat.
So for the next, I think if I could just decide and have my lab and do genetics, I would really like to have information about the sex and try to understand the sex-based dispersal. And I would like to expand the sampling. Having information about, for example, already just having sampling in Portugal, that is also in the Atlantic. We know that we have this Galician upwelling, see that is there structure, or just have more unexplained sampling to try to understand. So for me, personally, I would like to go for sex data and for expanded sampling.
Okay. My second question is, to your opinion, how could we translate these scientific results into conversation and discussions with the policymakers and the managers of the stocks?
I know that, for example, the project I'm working in, it's, as I said, we did all this in collaboration with fishermen, and they also have contacts with policies, and if we just look at what labels they can get for sustainability, if we have information about the genetic data, it's also showing, like we can have information about if the fishery is sustainable or not, and I know that like in Spain, they have those quotas, so it means that they know that something is happening. And I think it's just because there's this really big gap right now that we don't have communication with the policies. But I think that just already in terms of the shark remap project, they're in contact with fishermen first, but also with like policies in Asturias. So I get that it's difficult to convince people, but if we have data, it's already easier to say there's something that we have to be careful about than if it's just based on, oh yeah, we're fishing more rays, so it should be okay. But there's this labeling, so they're maybe not okay at all.
I agree with you, but sometimes I think that policymakers are not able to understand scientific data with this high level of difference. For them, honestly, because we are doing different job.
Yeah, I think outreach is important, but I don't want to be like advertising the Shark Remap project, but they do have a big part that is on citizen science and on outreach and trying to explain the importance of, I know rays are not sharks, I know people really like sharks, but they're also cool, and I think if people get to know them and want to protect them, it's also going to help. So this big outreach part of the project, I think, is a really interesting part that can, at some point, if we get the citizens, we get the policies. So, yeah.
Thank you.
Thank you, Silvia Gonzalo.
Okay, so thanks, Joanna, for such nice work. I really like it. So thanks for engaging us to the presentation, active presentation. So congrats on that. I have the pleasure to see you the way that you were working over here. So that's lovely to see that you're coming to finishing your thesis here. So I would like to highlight that I like the way that you started. You introduced the problem. It's not going directly to your species, but you introduce the problem, so congrats on that. And you clearly identify the knowledge gaps. And I think that it's really important thing to do, so congrats also on that. Yeah, so just some minor things that I have some comments and then we will have time to discuss later. So I think you focus on really local and really, I mean, you show really nicely the high distribution, the distribution that you have for that species, that rare species. So, but it's really local, your focus. Why that small in the distribution? Which is the main justification for that?
Getting the samples. So while I was doing my thesis, my supervisors were already trying to get samples for other locations. So in terms of timing, the sampling in Asturias is something that they do every year, so I participated in that one. But in terms of timing, we didn't have time to like go collect ourselves the samples, and also because they focus in Asturias and at least in the south of Biscay. But while I was there, I was really talking with my supervisor about, oh, maybe you're going to get in time some samples from Portugal or from UK. But now, during summer, I think technically for September, there should be samples of Portugal. We hope if we can get them, but of course it's science sharing and getting them from the UK. Maybe there's going to be something happening in the Mediterranean. They're not sure they're going to be able to catch any undulata, but it is happening.
Okay, so and then just more technicalities. So you were showing us that there's some lower than expected heterozygosity in the case of Microsoft. Have you tested for new alleles? Because since you are using the heterologous,
yes,
might be partial an explanation for that.
Yeah, so I didn't include this in my presentation because it's 15 minutes. But I did test for null alleles, and I got markers that were detected for null alleles as they were in other studies using those markers. And then, as I said, that I tried everything to understand what was going on. I did run all my analysis with the null alleles, the markers having null alleles being rejected, and the overall results were not changing, so we still kept them in. But it's something to keep investigating if this project.
But you have some microsoftware that consistently were a signal of neural needs
Yes.
for many different populations. And you
Yeah,
keep-
but the overall results were not changing,
Okay.
and based on other studies, we decided to keep them.
Okay. So I would say that I mean this clearly when you have this distinct signal is typical when you are analyzing mitochondrial DNA with nuclear markers with microsatellite. It's something that it's you expect somehow you do the effective science that you have. And the thing is, how do you test, because you didn't tell us from where those samples were taken, or were taken in the same season. Do you think that it's important to test the temporal, not only the local region, but the temporal? If that structure is really stable over time, or it's just stochastic due to effective population size, due to drift or whatever, at that time you take that, or if it's stable over time. I think it's really important in your case, because you have really tiny genetic differentiation between two very close localities.
Yeah, that's why I said that I would like to have, I think it's in my other slide, but to have seasonal patterns, try to explore that also because we know they have aggregation. Here, in the case of the Asturian samples, they were, and I mean also Galician because it's the Spanish regulation of having quotas that open and close fisheries. So they are on average kind of all in the same seasons. We have some extreme that could be, for example, the sampling happened when I was there. So from January to I think we finished in April, and it's like this every year. We have samples in Asturias that are for 3 years. The samples in Galicia were only collected over 1 year, but this is because this is what they had and it's better than nothing. So, but of course it would be super interesting to actually see, even with telemetry, seeing that if they actually aggregate at some moment of the year in specific places and trying to understand the biological meaning of the structure.
Particularly when you have really just some genetic differences, but not really strong differences, it's really important to show that that's stable. Because if it's stable over time, that is in your favor, saying that clearly there is something going on there. So that's all for me. So congrats again, great job. Well done.
Yes, we do have time.
Yeah, more like a curious question. Did you make, I think it was written, but your own drawings for the species of
Yes.
race? Yeah, very nice.
Thank you. Yes.
So you focus a lot on your population structure, but I think very interesting that you find inbreeding.
Yeah.
And have you can you link it with demography? Is that there are changes or there were changes in the last decades of this race in that area?
The thing is that there is no data on anything in that area about the race, so it's really complicated to know. There were some studies more in Galicia that were trying to see this site fidelity and localized aggregation. But also, even when we look at the landings in Asturias, actually there's a problem of the label, because in the past it was not labeled as Raja Undulata, so we only have Raja, so we cannot even have information about how it evolved with time. So.
Okay, thank you very much, Joanne. So now is the time for our second speaker of the second session of this morning in room 5. Just wait for your presentation to be shown on the screen. It's PowerPoint. Okay, so it's my great pleasure to introduce to you all Anne, aka Eddie. And Eddie is gonna tell us about the complexity and color enhancing Sydney rock oysters, Aquastrea glaberata, recruitment and reducing predation through substrate design. Eddie, 15 minutes, 3 minutes left. I will stand and let you know in case there is 1 minute left. The floor is yours.
Okay, hi everybody, good morning. Thank you for being here for my thesis defense. Dr. Luana already told you the title, but really think of it as me telling you a story about baby oysters navigating the treacherous waters of Southeast Australia. Okay? So my thesis project is supervised by Brett Stroud and Melanie Bishop. So here's the table of contents. I'll tell you a little bit about shellfish reefs, talk about the Sydney rock oysters, the ones I worked with, the study sites, and with this, there are 3 different experiments that I ran for to study the color and complexity, and I'll do conclusions and references. All right, so shellfish reefs, what are they? As the name suggests, they are made of shellfishes. So they could be things like oysters or mussels, forming like 3D aggregates. And with this, they form like reef structures that provide a habitat for other organisms to live in. They are like a good nursery habitat. They're also really good for reducing wave energy and stabilize the structure. So they often occur in conjunction with other habitats like mangroves or seagrass. So as you'll see from pictures later that I'll show, like the ones that I've been using, it's occurring next to like mangrove forest. So one thing that's been happening all over the world is a process called coastal hardening. So as cities expand, like a lot of the natural coastline is replaced by hard structures, like these ones over here. So these are often structures that are flat and featureless, and they don't support as much of a biodiversity as a natural habitat would. So like what it is, it's like with this, there have been projects that have been occurring that are aiming to retrofit them with panels that kind of increase the habitat complexity and make it more suitable for other organisms to live on. So this one here is a picture of the Living Seagulls project that's happening in Australia right now. And with that, they have different panels of different types of complexity suitable for different types of organisms. But with my project, I'm specifically looking at this little guy there. It's the Sydney rock oyster. So as you can imagine from the name, they occur in Sydney. These guys used to be quite widespread in Southeast Australia, but now, like because of historical overharvesting, they're like left with maybe about 10% of their original like distribution. So the historical harvesting methods are really destructive. They dredge the bottom, so a lot of the like original structure that's there gets destroyed as well. So here's a very simplified picture of a regular oyster life cycle. Like they often have a pelagic phase, and the larvae will be like swimming around looking for a suitable place to settle. Then they would like attach themselves to the substrate, and then they'll grow to become adults. So with the shellfish reef, like for them to really like make an impact, they need to grow big enough to have this like 3D structure. So for my project, I'm looking at specifically the early life stages of the Sydney rock oyster. So with this, I come up with my research questions. As I mentioned before, there are already previous projects looking at panels and different substrate types or like different orientations and how they influence recruitment. But not many studies have been looking at color. So we were looking at substrate color, complexity, and the size of oysters and whether it influences the recruitment and survival of the oysters in urban estuarine environments of Eastern Australia. So you can see my research questions there. On this map here, I have the study sites. So this area is where Sydney is, and the 4 different pink dots are the sites for my recruitment study, which I'll explain more later. And the green point there, it's the site for my predation study. I have 2 different experiments run there. So if you're wondering why they're very different, it's because in regulation, for my predation study, I use aquaculture oysters, and they are not allowed to mix estuaries because of a previous disease. So the first experiment, recruitment experiments. So these are the 3D types that I worked with. With the recruitment experiments, I was looking at complexity, so whether it's flat or it's complex. So as you can see on the left side there, those are flat panels. Basically, they're featureless. They're flat 5 by 5 panels, and they have a surface area of 0.065 meters square. And these ones here are the complex panels. They have 5 centimeter ridges, and like as you can see, they have different areas for like things to recruit. They have a bigger surface area, as you can imagine, at 0.136 meters square. And for the color side of things, I have 4 different colors: red, green, gray, and yellow. So in total, I have 8 treatments, and there are 5 panels of each treatment and 40 panels per site. So that's 4 different sites. Okay, so this is how it looks like when it's installed at site. Like basically they're randomly placed onto a seawall and drilled in. So they're standing there on the seawall. They're left there for 4 months to check for, and I'll come back after 4 months to check for recruitment. And I'll look at the number of recruits, the location of recruitment. I forgot to mention this earlier. So for the complex tiles, because they are like ridges and crevices, so I differentiated where they were recruited as well. I also looked at the length of the shells. So live recruits are defined as new individuals with both valves attached and non-gaping. This is an important factor because with recruitment, there is a mixture of effects between actually recruiting and then not being eaten after it's recruited. So then it comes the stats part of things. So for the first test, we used abundance data from the settlement panel pulled across the sites because there was an error that happened during data collection. So we had to pull the data and use each site as a replicate instead of like, you know, looking at individual tiles. Unfortunate, but it's okay. So I used a GMM and there was no significant effects at all. So we didn't find anything, even though it looks like this. Yeah. So we decided that, you know what, like maybe since we have the surface area of the panels, let's standardize the abundance data to the surface area to make sure that like the effects that we might see or might not see is not a function of the surface area. So with that, we standardized it to per meter square, and we ran another model. We dropped the site effect because, as I said, we don't have replicates for site, like that became our replicate. So we found an effect of color. We did pairwise comparison tests for all the ones that were significant, and we found that gray and yellow panels had more recruitment than red ones. And then complexity, as you would imagine, like complex panels had more recruitment than flat ones. And with the interaction, green and gray complex panels was actually with more like recruits than red complex ones. And then like with the data that we had, the full set, like we ran separate analysis for each site. So I looked at the Ramsgate site and the Sanssouci Park sites individually, like separate from all the other sites. And we ran 2 different tests, one with like all the panels, and another one with just the complex panels, trying to like tease out the effect of microhabitat, which is the crevice or ridge. So with Ramsgate site, we didn't find any like significant interactions from any of the above. But for the Sanssouci site, we found like effect of complexity again. We also found that like with the complex panels only, there was an effect of interaction where the green and like green panels that has crevices had, the other way around. The green panels had more recruitment in crevices. Okay, so I know there's a lot of information, but in summary, there was a clear effect of complexity. Like that has been in line with other studies that have been done before. This is mainly could be because like it buffers them from heat, desiccation, provides protection from predation. As I mentioned before, it's like a interaction between 2 different like things that influence recruitment. Past studies have also shown that like larvae are negatively phototactic, which means they like dark spaces. So they would, with a crevice, you have shade, they can go in there and get recruited. And something that's interesting about study is that we found that red complex tiles actually had lower recruitment than the others. But past studies said that like, oh, oysters actually like red surfaces. So this might, we think that it might be because it's actually a different species. They use pearl oysters instead of the Sydney rock oyster. There might be species differences here. Also, like, with the interaction, it's not super clear. There might be like different, it might be because of like the visual cues being hard to detect in high turbidity areas, like urban estuaries. So like with the 2 different sites earlier, you can see that the graphs were quite different with the recruitment. I think with this we can kind of tell like abiotic factors at different sites is possibly like also playing a factor. So moving on to my second study, it's a predation experiment looking at size plus refuge. So for those of us who don't know what size plus refuge is, it's basically to see whether as a creature grows bigger, whether they are safe from predation just by their sheer size. So with predation, a lot of like predators are limited by the size of their prey item because of how big they can open their mouth, basically. So with this, we designed an experiment where we glued the aquaculture oysters onto the stakes, and I had 5 oysters per steak and 7 steaks of each size class. So the size classes are listed here, and we had 1 control for each. So when I say control, they're basically caged treatments. We made a mesh that excluded the predators from being able to access the steaks. And so as you can see from the picture there, all the steaks are stabbed into the mangrove site and with natural oyster reefs. And they're care was taken to make sure that they're below the high tide line when the tide comes up. This is to ensure that the fish has access to the oysters. Another thing that we did was also to make sure that they're not actually super close to the bottom of the floor, so they're excluding things like maybe crabs that can access them as well. Okay. So with that, we got our data. We found that of all the oysters that were uncaged, 11% was alive, but most of them was cracked or missing. So this is a very good indication of predation pressure. So you might wonder why is missing also part of it, because in our control experiments, in our controls in the cage, we found no glue failure. So the only possibility is a predator coming in and grabbing them away. And they're actually very vicious. They would actually even grab the epoxy off. So you're running the data analysis with the uncaged steaks. We found that only the large size class was significantly significantly surviving. And when I say dead, I meant missing or shell cracked. So this is consistent with previous studies where they looked at predator game size, and also interestingly, even though it's not significant statistically, we found that some of the small size class was left uneaten. And this is very unusual, something that I thought we should maybe explore a little bit more. I actually found previous studies that they mentioned that it could be because they're less energetically rewarding for the predators to eat. So the effort used to predate on them was not as high as what it would energetically get. And we actually took some of the extra small size class oysters and we measured the flesh to shell ratio by using dry weight, and that's actually what we found. So this is just a picture to show you what I meant by cracked shells. So you can clearly see that they're cracked and they are basically bite marks. So for the last experiment is the color complexity refuge. What it is I used the same aquaculture oysters, I measured all their sizes, and I glued them onto each of the individual panels. There are 30 oysters per panel, 15 on ridges and 15 in crevices just to make it balanced. And then we placed all of them in the same mangrove site as the steaks, and we left them there for 2 weeks. So similarly, I also have cage treatments and uncaged treatments. This is basically acting as a control to check for non-predation related mortality. And also with the uncaged treatment, as you can see in the background there, this is how a natural oyster reef should look like. They should be forming 3D structures that could naturally, if you think about it, protect the oyster recruits if they get recruited on the shell. So what happened was this was before. And then we placed them in the field, the fishes came for a buffet, and then after 2 weeks, there was absolutely nothing left. They ate it all. If you look at the red arrow, this is where, this is a bit of epoxy left, and so this is them just grabbing the oyster and going. But if you look at the blue one, this is super interesting because they even took the epoxy off. Very hungry. So then after that we ran the data through a linear model. There's a very obvious effect of the caged and uncaged treatments, and I can even see it from my eye because with uncaged ones, they were completely eaten, and with caged ones, even the flat ones had full survival. So this is kind of obvious, and it really shows there's a strong predation pressure in the estuary. With the uncaged panels, we ran extra stats analysis on linear models. We found that complexity really helped improve survival of the oysters. And this is just an observation, but all survivors were found in crevices. So we found that substrate color had little effect on oyster survival from predation. This might be because the estuary where I was working in, initially we wanted to put cameras in, but because the water was so turbid, you can't actually see anything from the camera. So we decided that, you know what, let's just use other cues like crack marks and stuff. The physical barrier of being structurally complex reduces the detectability of the oysters to the predators. And so here are some of my key findings. I think one thing to note is that once oysters reach a certain size, like they actually exceed the predator gauge size. So this could mean like in the future that like we could protect oyster reef projects to up to a certain size, and then it might improve the success of the project. So the color effects, even though they are subtle and consistent, they might be present, but more tests need to be done on other areas as well. So we also wanted to look at other shellfish species, extend the time of the study, and maybe look at other environmental factors. Okay, that's it. Thank you.
Such a big, ambitious, and interesting project in a little bit more than 15 minutes, but it was very clear, so thank you. Any volunteers?
I can start if you want. Thank you for your presentation, super enthusiastic, and really clear, and really good job. We have a couple of questions. I think that your job, in addition, is super useful in the framework of habitat rehabilitation and oyster reef restoration. So it's super. I just a question. Why focusing on color instead of typology of materials, for example, or other characteristics of the stars?
Yeah, this is actually a really good point. Like, actually, a lot of past studies have looked at things like material type, complexity, the shape that you make, whether it's a pit or like, you know, whether it's just like little grooves, even like micro habitats, like maybe 3D printed things. Color is just something that's not so much done. And personally, I also feel that like with the color differences, if we do more studies on it, we can also relate that to the predators' visual systems and eventually have like a fuller picture of like how, you know, like they can survive better. Like it's also a low cost thing, like just by changing the color of like the eco-fitting panels, like eco-engineered panels, like if that can improve recruitment, why not? Yeah.
Is there any biological hypothesis on which you can hypothesize an effect of colors on the recruitment?
Yes, there are other, like, these panels are actually also used for a wider study, and the panels are used to look at recruitment in general across the board. So like with that, I was specifically looking at oysters, so I used the same panels. It was kind of like by chance that they are also recruited on these panels, so then I wanted to also look at color effects on top of that.
Thank you.
I was wondering, so you got, so you changed the effect of color, and I was wondering if possibly, because I don't know too much about the visual apparatus of an oyster larvae, but I was wondering, do you think that the different chemical makeup of the paint, rather than the color itself, could have played a role?
Yeah, definitely. I do think so. So to counter that effect, like actually all of the panels before we put it into the field were coated with pond sealant. Okay. So that all of them were actually equal. Yeah, so one thing I also didn't mention is that all these panels actually have the same brightness, they just differ in color. So then brightness is also not a factor. Yeah.
I have maybe one more, a curiosity question. Because coming from Brittany, when you mentioned that for oysters, you know, the months of the predator is important and all that, I was like, what? And then you showed the predation by fish and all that. The thing is that in Brittany, oysters are rarely predated by fish. The main predator are tiny snails that pierce through their shell.
Yeah.
And I was wondering, do you have those as well?
No, so with my study, because it's only 2 weeks, like it wasn't maybe there long enough for the snails to come in and like get to the oysters. Okay. And previous studies have been done on the same site, and the fishes are very hungry, like they just come right in. I almost feel like with my experiment, I should have maybe reduced the time of leaving them there just to see like it happening, you know, instead of coming back and realizing, oh my god, they're all eating.
Okay. Yeah, I guess related to that, I feel that was pretty much well controlled, but, and I guess that was in mangroves, right?
It's like a mangrove site that has an oyster reef as well.
Yeah, so you don't have a lot of, I guess there's not that much hydrodynamism in there. But you could,
Yeah, it's very calm, it's like a low energy site, like, you know.
No, it's in terms of possible arrangement.
Yeah. And we also took pictures, and the tiles were exactly where we left them. Yeah.
Okay, I've got another maybe more technical question. So you had one of your tests, you, if I remember well, you didn't find statistically significant effect, but then you standardized to area.
Yeah.
And then you found effect. And I was wondering what kind of area did you use? Because
I use
and how was that?
Yeah. So when I say I standardized the area, it meant that for the flat panels, I divided all the recruits that we found with the flat panel surface area.
Yeah.
And then with the complex ones, I divided it with the flat, like complex area.
So you include the grooves and all that?
Yeah, I included the surface area of the grooves as well.
Okay. Do you see a potential problem with that in terms of your analysis?
In terms of like using standardized data?
I mean, this way to standardize them.
Yeah, yes and no. Like, I feel like it's, if anything, it should standardize things better because it accounts for the fact that the complex tiles has more surface area. But like it also means that like the complex tiles would always kind of like have a lower count, not lower count, but like kind of lower, they're like biased, I guess, you know?
Yes.
So that's the only thing, but I feel like the benefits outweigh like.
But so then, why do you see, this is what bothered me a little bit because I agree, but why do you see no effect when you don't standardize and you standardize and you do something that should increase your, that should decrease your effect, and you see an effect?
Yeah, it's a very good question. I asked myself the same question. I don't know. I will think about it. The study is actually, we are collecting more data for the study. So there's actually another round of collection of data, and we're going to use that to run the stats analysis again to better understand the influences of the surface area and the complexity of tiles on recruitment. Yeah.
Okay, so first of all, congrats for your work. I really like it. I really like manipulative experiment. I think that even though it's not the right, in some cases, representation of what is going on in nature, help you to understand many things. So congrats on and I think most of all your experiments were carefully thought about and controlled. So well, I really enjoy your job. So how maybe you mentioned, but I forgot. Why did you choose those particular colors and not others?
So the 4 colors, these are, like I mentioned, the tiles are used as part of another recruitment experiment. And the colors were chosen by that previous study to mimic like colors that you can find in the Sydney Natural Harbour. So like things like, you know, sandstone, rocks.
Maybe I missed that. I'm sure you mentioned that. I just So one thing that I was wondering, have you, it's clear the related on that, you didn't find any difference, I'm talking about the second or the third experiment, you didn't find any difference in terms of color, no? I mean, all of them were, So, but what did you control for density?
Density of the number.
So how many oyster per square meter do you have? And that was similar to what you found in the beach? Because could be like you were feeding the
yes,
fish.
we were feeding the
so
fish.
It's not really representative of the density that you have in that particular location of the beach, and then that needs an attraction on them.
Yeah.
And the thing is that you didn't find a difference between a coral. I mean, all of them were more or less equal because what you are finding is critical to that graph.
Yeah, of course.
That the one that the access in the crevices. It's more difficult. That's why you have a remaining 15% or something like that.
Yeah.
Not really. So it's a I'm understanding
Yeah,
correctly.
yes, that's
Which
right.
was how much? Which was the difference in terms of density that you have in your panels in comparison with
The natural.
With the natural? Do you have an
No, we
estimate of
didn't
that?
actually go to the natural side and sample the actual amount of juveniles of that specific size.
Yeah.
But
it's hard. I guess that it's a lot of work. It's a lot of work.
it's also kind of hard to find them. Like in a natural oyster reef, like you have to sample a small area for a very long time, just because they're visually difficult to see, right? Like oysters would settle in crevices that are hard to reach. So that if we were to do that, like it would not be a master thesis.
It would be really informative to repeat these with two different densities, one almost similar to the one that you are finding more or less in that region of the beaches. Yeah. And another one a little bit higher but intermediate to that, and to see and then to plot and see if that change or not. I think it's
I think so. I think
really
that
exciting.
could be interesting for future studies. Yeah.
That's really nice. And what else? Yeah, so no, that was mainly about the density thing, but in any case, I congratulate you. So well done. I really enjoyed the work. So
Thank
well
you.
done.
We do have time for one, maybe two questions from the audience. If not, congratulations again. Oh, we do have, oh, I'm sorry. Please, go ahead.
Very quick question. You mentioned that there was some investigation into the material in previous experiments, and you said that you wanted to focus more on color. But the material of the panels that you had, I'm sure, I don't remember, what was the material and was there a correlation to the past experiments to also look at your experiments?
Yeah, so the panels that I used are actually concrete panels. The colors are mixed into the concrete, and then they're, the flat ones we made it in-house, and the complex ones, they made a mold with a lab in Melbourne with a 3D printer, and then they used concrete to pour into the molds and made them. So past studies have shown that like oysters are okay growing on like concrete, but they prefer things like shell or like natural rock.
Thank you.
No worries.
All right. Last and definitely not least, it's my great pleasure to introduce to you the third speaker of the second session of this morning, Sofia Maza Villarreal, who is going to tell us about, from a shark's eye view, automated shark detection in animal-borne video. Sophia, remember, you have 15 minutes. When there is 3 minutes left, I will stand up and we will also signalize there is 1 minute left. Success, the floor is yours.
Thank you. Thank you, just as Luana said, I'm going to speak a little bit about from a shark's eye view perspective and do automated shark detection in animal-borne video. So a little bit of what we will see today, background of the study, introduction and aims, data and methodology, results, discussion of future directions, and of course, conclusions. So a little bit of why this is even important. So as we know, sharks are predators that allow us to help us to shape the reef ecosystems by controlling the prey behavior and the community structure in a top-down effect. Specifically on the side that I'm going to talk about in my study, which is the Fakarava Channel, specifically the South Pass, it is well known as one of the largest shark reef aggregations, approximately 700 reef sharks, which basically this is seen nowhere else in the world. So this is really interesting site to study the whole fish community structure, how sharks behave, and why is there a shark aggregation there. There's already a little bit of literature that shows that this specific community of sharks show a daily aggregation in the current zones. So they take advantage of the ingoing and the outgoing currents, and they exhibit coordinated group hunting at night, also following a little bit of the white-tip sharks that are around that zone. But studying sharks is not easy, and the traditional survey methods like divers or BROVs introduce constraints and biases. So to break this constraint, this study utilizes animal-borne cameras. Why? Because it allows us to have a first-person footage from the shark's perspective, which makes it unbiased and continuous, and it also allows us to see if the sharks go into a cave, which, for example, a brook would not be able to do. It allows us to capture the natural movement and the interactions of the sharks with every other individual that's in this location. And of course, we can see the habitat selection of the sharks. And like I had said, Whakarava Channel has a big aggregation of sharks, so this is ideal for dense aggregations of sharks. So imagine a diver trying to count 700 sharks all at once in an hour dive. It's impossible. So there is one constraint about animal-born videos. It creates a lot of raw data. That basically it's very labor-intensive and very expensive to be able to manually analyze all of these data. So that's where deep learning models come into play. So a deep learning model has a technique called machine learning, and the machine learning that I utilize is called YOLO, you only look once, and it allows us to automatically detect and classify the sharks. Also, once we are able to do this, we're able to extract usable metrics like, for example, max N, and it allows us to eventually reveal ecological patterns across the depth, the habitat, the type, and any other environmental variables that we want to see. So for the objectives and the research question, my main objectives were to fine-tune a deep learning model, YOLO 11. Like I said, you only look once, and 11 is the version that I used, is the newest version, on a custom dataset. So a dataset from the Whakarava channel. And we wanted to test whether the model can reliably detect and classify sharks from the animal-born footage. And then we came up with the research question, can deep learning automate shark monitoring from animal-born video in a way that supports ecological research? So like I had said, the study site is the Whakarava channel in French Polynesia, specifically in the South Cast. It is a UNESCO biosphere reserve, which makes this study even more interesting because it's a place where it's protected, there's no disturbance, and so we can see the natural movement and behavior of the sharks. The collection was done between June to July 2017 by my supervisor, and back again May to July 2018. And the study setup was as shown in the picture. There were 4 HD video loggers mounted on 4 gray reef sharks on the dorsal fin of the sharks, and these tabs were attached via dissolvable galvanic links that made it easy for recovery. So once I got the data looked a little bit like this, and I got approximately 82 videos of animal-born cameras, in which I was able to detect 4 different shark species and also detect 3 different environmental variables, type, depth, and habitat in a categorical way. So now, for the methodology. So first we're gonna go into the raw footage. So our raw footage, I randomly selected 21 videos plus 11 supplementary clips that were recorded by a diver under the same conditions at the exact same time. And then these videos, I had to do something called a process of annotation of videos. So the actual process is shown in the video here, but basically I utilized an application called wave-based annotation of videos for ecology. I segmented the videos at 5 frames per second in order to not lose a lot of data, but also not waste a lot of time annotating my videos. And then what I collected was the species ID, the shark proximity from the animal-born camera, the depth, the type, and the habitat. And approximately I had around 4,400 annotations across the 4 shark cameras, and approximately 2,000 fish annotations. And so basically one annotation is one bounding box. One bounding box is one of the squares that has an individual inside. So imagine the amount of data that I had to go through. So after this, I had to still do an extra step before I was able to train my model, which is called the data preparation and the split of the dataset. So the data preparation was a normalized format from the wave bounding box, so the application I used for my annotations, into the YOLO bounding box, which is the format that I use for my model. The training dataset, YOLO Ultralytics, recommends you to split it between 80% and 20% for validation dataset. So basically, in the training dataset, you're telling the model what to study, and in the validation dataset, you're asking the model, what did the model learn? And we decided to do 2 different models, one for species classification, which will give me the species name, and one for shark detection, which will just allow me to see how many sharks in general as an individual are. After that, I took it a little bit further and I went into an active learning loop in order to have even better data and a much more accurate model. So I first went into transfer learning, which is an already trained model to detect general objects and then I adapted this into sharks. And in order to have even more data, I utilized data augmentation, and I utilized these specific 3 techniques. There's many, but I chose just these 3. So first mix-up, it takes 2 images and their labels, and it interpolates them together in order to be able to distinguish sharks in any type of environment, at any lighting, or in any position, and to have a better generalization and not just choose and pick which class ID the model identifies. Then scale. Scale zooms in and out of the images, allowing the model to really detect the sharks in a reef natural conditions. Because of course, if you're looking from an animal-born video, the shark can be very, very far and it can look very tiny, and this allows the model to detect that shark, but also a shark that's proximity to the animal-born video. And finally, but not least, mosaic, that puts 4 images together as a collage, and this allows the model to detect any class that we have without having class imbalances. And now we actually go into the active learning loop, which an active learning loop is basically you ask the model to take unseen and unlabeled frames to make predictions, and then I get the predictions, and then I review them, and they go back into the data set of the training. So for example, here we have a correct label. This label, I would not touch it, and it would just go back into the training data set. And here we have 1 correct label and 1 incorrect label. Here I would correct the incorrect label, which is, here it should be Tangarinos melanopterus. I would change that, and it would go into the training data set. And I had a total of approximately 776 annotations, which allowed me to have better accuracy for my model. So finally, I have something called the prediction results. So basically, the prediction results is my video showing the shark detection model and the shark classification model, and this allows us to visually see that our model is accurately checking sharks in the animal-born video conditions. And this was tested on a video that we did not utilize for the annotation of the data, so we did not have any bias there. But now, I will show the actual results of the performance of the model. First, I'm going to speak about the shark detection model. So for the precision recall performance, as we can see, our curve is very high, and we have a mean average percent of 0.96, which that's really good. That's telling us that basically 96% of all of our data is being classified as sharks, which is correct, and only a tiny 4% is being missed, which is right here. So here we can see in our confidence matrix that here would be the true, that's the predicted, and so here our model is predicting the background correctly, it's predicting 96% for sharks, and it's missing 4% for sharks. But for the time restriction that we had, this is a really good result, and we can even see it with the F1 confidence curve. Here we can see that anything that we put a confidence threshold above 0.62, we're gonna get a confidence of 0.91, which that's really high, and it's higher than most models that are available nowadays. And now for the shark classification specifically, we also have really good results. Our mean average above all classes was a 0.88. That means that 88% of the time, the model is going to classify our classes correctly. And you might be wondering, why do we have 2 classes that are even higher than the general class? And this is because these 2 sharks have a specific thing that distinguishes from the other sharks. Carcarinos melanopterus is the black tip shark, so of course it has a black tip. And Triadonoversus has a different snout shape and a white tip, so of course it's easier to recognize. But here we can also see it in our confusion matrix. We had really good results, although there was a little bit of confusion between the background and the shark, and this is due to the fact that we have to remember that it's an animal-born video, so it's constantly moving, so we have a lot of frames that are blurry or the light changes, and the coral reef structures in this location, it's very big coral reef structures, so there was a bit of confusion there. But there's still, some things that we can improve, and still our F1 confidence curve was quite high, allowing us to still have a really good score of 0.84, which in 5 months, that's great results. But we still decided to try to make sure that everything was working, and we did something called max n. So max n, we did maximum number of individuals observed in a single video frame, and this, I manually counted them. So I manually counted 26 videos, approximately 1,632 manual annotations, and I compared this with the shark detection model in order to see if we had a good correlation, and we had an R-squared curve of 0.88, which that's a really good correlation. We do have some outliers where the model outperformed or underperformed, but again, it's an animal-born video, footages are confusing, and even for the manual observer, it's quite hard to try to count 100 sharks in one frame. So we wanted to show an application of ecological results. So this is not the latest result, this is not what is going to be published in the article, but this is just to show an application of why this study is important and why should we use this study, right? So in this graph, I'm going to show the results for Shark Camera 3. Here in red at the top, we have the Shark Max N on a function of time. This is the real time when the video was recorded. And here on blue, we have a time series of depth, habitat on green, and tide on orange. And just by observing this, we can see that most of the aggregation of sharks is earlier in the day, when it's deep depth, coral dense habitat, and incoming tide. But when we compared it to the other shark cameras, there were no real spatiotemporal patterns, and we also did a GLM plot, and we had no significant values. So this we still need to keep investigating and trying to get better results for this part. So just what worked. So as I said, we have a high detection accuracy for sharks under real reef conditions. The animal-born cameras were able to capture footage that divers cannot, so that was one of our main constraints. And the model was able to handle turbidity, clutter, and lighting shifts, which normally that's one of the biggest issues with deep learning models. But of course, what needs improvement? Manual annotation is a bottleneck. It takes too much time. It's super slow, it's exhausting, and it's expensive. And because of that, we had very poor fish detection and a very small data set, which caused a class imbalance. So we were not able to include fish into this and have a bigger perspective and a bigger picture of how the fish or community works in that area. And the current pipeline only works for the shark detection and classification, but we have not tested it for behavior or any other interactions yet. But it has a big ecological value. So this is a non-invasive technique, and it can be continuous and scalable, and not just for our project, but for other projects, specifically right now in the Pacific region. But eventually, this can be expanded worldwide. So what do we want to do for our future directions? Like I said, we want to expand the dataset with more annotated images, especially of the fish community, because I was able to identify 93 different species of fish, but still, we did not have enough data to analyze it into the model. We want to include more data augmentation and more active learning to reduce the manual label load. When the collection was done, there was also a logger data, so we want to add the information of the logger data to improve ecological context. And last but not least, the model will be open access for the public in order, if you guys have a video of sharks and you want to identify your sharks, this model will be available for the public. And it is a scalable tool for long-term monitoring. So just to conclude, this study is really important because first, it supports a scalable long-term ecological tracking. It's a non-invasive technique, and it allows us to reveal patterns of shark gregariousness in locations where, one, it's really complicated to do it as a diver, two, it's really complicated to do it as a pro because you introduce the bias of the fish bait, and this fixes all of these solutions, and we get good results. Thank you.
Thank you very much, Sophia, for this excellent presentation. I'm sure everybody found very interesting. And I'm not surprised that you took this challenge of videos in the live. Okay, now for questions from our jury members. Gonzalo, would you like to start this time?
Yeah. So first of all, I would like Sophia to congratulate you.
Thank you.
So you did a great job in the presentation. And really, it's a pity that I cannot read your manuscript. I really would like to do it. So
I can send it to your way.
Yeah, but what I would like to highlight is how complex are all the methodological aspects that you did, and how well you explained for us that it's the first time that we have listened to you. So that's not easy job. So well done.
Thank you.
So thanks for. Also, one thing that I would like to highlight is apart from using this deep learning, how well it's designed using different kind of internal controls to see and to check how it's the performance, how you're improving the performance, but how you're using control data to see what is going on. If not, it's actually like a black box, so that's also covered to that. This is just out of curiosity, it's not my expertise at all, but did you have the chance to feed this, the software or with some video from a different location but in the same region, and what was the performance?
Yes, we did. We actually even tested it, I tested it for curiosity with a video that I had from diving in Mexico. So it's a completely different region and it worked. So that's why we were even able to say that our results are very accurate. We also compared our results with another publicly already available platform called Shark Pulse. And our results performed a bit better than theirs, to say it, but there is, our model is still not well prepared because it's only detecting the accuracy of 4 species, correct? So we need to amplify that in order to be able to test it for multiple different shark species. And we also compared it to the same region at different depth that my supervisor had videos of that he took diving, and still the model performed really well.
And if you on purpose add videos where you know that you have a really complex assemblage of sharks that are not from those species, have you tested how they perform if it's moment of the...
From different species, you mean?
Yeah, from different species that you
the model basically detects shark, and that's it. As here, I don't know, So here, as you can see, we have sharks, and that's because the model is not confident enough to detect Carcarinos ambrivencos, which we know because of the literature cited that it's Carcarinos ambrivencos. But it's detecting shark because we're telling the model, unless you're very confident, go directly to shark. So that's how we fix that issue.
Yeah, and so I think you mentioned that with the same with changing light. So it was the same if the video was taken in the morning or in the late afternoon.
Right. So our videos were not recorded at the same time. So we have videos that start at 4 a.m. in the morning, and they finish at noon. And we have videos that start at 2, 3 p.m. and they finish at 6, 7 p.m. We do have a little bit of a problem still with the night part because we were recording, the recordings were done with GoPros. So the GoPro is not the best quality camera, but eventually with better quality videos and with lighting and everything, this can be done, and it can be at any time of the day, and the results are still the same. They don't change.
Thank you very much, congrats.
Thank you.
Thank you, Gonzalo.
I have a question.
Yes.
Why deciding to mount the camera on a moving animal instead of on a fixed infrastructure, fixed on the boat?
Right, because if you do a fixed camera, it's similar to doing a broth without the fish bait. And so you only have the video of that specific location, and it gives you the constraint of you cannot see the movement, you cannot see the behavior, you cannot study the hunting, for example, the hunting behavior. The literature cited that I based this project on was done by my supervisor, Joan Morea, and he has studied that the group hunting of these sharks is very different than what a normal gray reef shark would behave. They follow the white-tip sharks into the caves to try to take advantage of the white-tip shark catching the prey, and then they steal the prey from them. If you do a static camera and you do it far away from that, you're losing that information.
And does the camera has an influence on the animal behavior?
Right. From what we can tell, no. This study was part of a project, a big project called Rapport Gombesa, which was done in partnership with Andromeda Oceanologique. It's a very big expedition. And the researchers that were there collecting this data were there for many, many months. So they tagged the sharks, and they were still able to see the sharks with the tag cameras. The sharks were behaving totally normal. So we don't believe. Yeah.
Thank you, Sylvia.
Yeah, I think it would work. I guess I got start with some small technical questions, and then I guess we can try. You mentioned that you have a matrix, I think it was, the
This
spark.
one or the confusion matrix?
No, a matrix, not a matrix, of proximity. But,
Yes.
And I was wondering what that was confusing.
Yes, so we annotated it. We had the initial idea of studying if there's a behavior difference between a shark getting closer to the shark with the camera or farther away. We were not able to test that due to time constraints.
Okay.
We did not expect to take this long to have an accurate result for the shark detection and classification, and we did want to take it even a bit further and do, like I had said, the fish identification as well, and the distance study as well. We have not been able to test that yet, but it's part of our future directions, so we can have a very big, complete picture of everything that happens in that specific site.
But so basically, it's just you said it's annotated, so it's
Right,
just
so-
you're like, this is closed.
So it was done as with 9 different categories. So we had, like, if the shark is on an angle, we would say angle, close, angle, middle, angle, far, or like side profile, close, side profile, middle, etc. And they do have a lot of measurements of many of the sharks in the region. So having the measurements of how long the shark is and the dorsal fin, we were going to be able to detect this distance, but we sadly did not have enough time to do that. Yes.
Second question, then you have kind of, I would call it a calibration line between, so actually, I think it was Maxen that you counted and Maxen that the model counted.
Yeah.
And so it's really nice, really nice correlation. But I noticed that it's not exactly on the 1-1 line.
Yeah.
So some either with the model or you count that, so someone counts more than the other.
Yes.
And I was wondering if you had any idea of why and...
The model counted more than me.
Okay.
And yeah, and I do believe that is one of the reasons why manual counting is not the best way to do it.
Okay.
I was exhausted when I was counting this. So more than likely I did the human mistake of undercounting sharks. And like I said, I'm not sure if you can tell, but for example, if I show, I'm just gonna show like the prediction video. If I show this and I pause it in the middle, and well, I cannot pause it, but if I pause it here and I tell you count, you can't tell that's a shark.
Yeah.
Like it's hard. The shark's over here, and then identifying them.
So that's you on the left and...
No, that's the shark detection model, that's the shark classification model. Both of these are the results of the model.
Oh yeah.
So the model performed even better than...
But I'm wondering, did you try, so those, for those specific sharks that were detected by the model but not by you, did you went back to the images that were actually detected as shark and were they sharks?
Yes, we did that, and we also try to, we have so many videos of this because you visually have to first see how the model is performing, and the model was not trained one single time, the model was trained more than 32 times, changing learning rates, learning curves, changing a lot of different parameters, and every time, basically what I had to do was watch these, manually count them, make sure the model was not overcounting or undercounting, and then the model gives me an Excel with the class ID and the confidence threshold that the model is throwing, and the ones that were lower than 60, I would go back and check them and make sure that they were the correct species and that it was a shark.
Okay. And I guess to a more general question, I understand it's not precisely the work you did here, but a lot of things already, you just like fine-tune the model. But I was wondering from those videos annotated with the model, what kind of ecologically relevant metric you could have from those, and how would you compute them?
Right.
Do you have some ideas? I mean, that could be a wide subject.
Yeah,
so...
there's so much. Like I said, it's UNESCO Biosphere Reserve, and it is not really permitted for like tourists to go diving there or anything. It's only for research. And so what's very interesting, and one of the reasons why these like wall of sharks happen is because there's also a big aggregation of groupers in this region. So that is one part of like how this study even started. My supervisor was studying the aggregation of groupers there, and they saw the big wall of sharks, and they are intrigued to study that. So first, sorry, I just went. So this is like the map of the study, and this is specifically the point on where we recorded those videos. The sharks don't go here. The sharks stay in the South Cast, taking advantage of the ingoing and outgoing currents. So that's one of the metrics that we can take. There's also different
you mean position?
yes, and there's also different coral structures, and the sharks either stay on like a channel where there's like a wall of corals here and a wall of corals there, taking advantage of the tide, or they are going into the coral structure, chasing the white-tailed sharks. So that's another environmental thing that you can take, like the whole habitat structure and the seafloor and everything, how all of that affects. But part of what also, the data is already collected, it was the speed of the shark that had the camera, what speed the shark was swimming at, what depth was it changing, of course the temperature, but the temperature doesn't really like, change that much. And yeah, that's it.
I guess maybe the last one. Are you able, or do you see a way? I have no idea. I guess it's possible because I think a colleague that did something similar with insect images. But are you able to, from the models, to follow maybe a new village?
Yeah, tracking. So we haven't tested that because in the beginning we really did not like come up with that, and we came up with that too late. We do want to see the tracking in order to also take the constraint out of maybe we're counting the same shark 50 times, right? And so that way we can have a better abundance number and like a more accurate abundance number as if we were starting to track the sharks, but that would be a totally different training that we would have to do.
Okay.
Maybe one quick question. If they are quick. Yes, super quick. I really like the presentation. Thank you. I was maybe not very quick. Are there like blind spots when you mount the camera? Like, I mean, I guess it's the future word for them.
Yeah, so there are, and sometimes we can even see a bit of the head of the shark where the camera was mounted. And that's why here, here as you can see, I have something called camshot, and basically that was me annotating for the fact that I could see, I don't have an image to show, but that I could see the face of the shark, so the head of the shark. So anything that was going under that area, I was not going to be able to see it, of course, because the only thing I could see was, yeah, and that's why I annotated it to see how we can utilize that information later.
Yes, I'm also interested in the camera itself and how was that detached and how long did it stay on the
Yeah, so, 1 second. Well, actually, I can show it later. Like, it's literally like this, and it's attached with a galvanic link that's dissolvable. So after approximately 4 hours of video, that link is dissolved and it goes into the surface, it floats, and then there's a GPS to go able and to be able to recover it.
Okay, and how, like, did they do it free diving? Did they catch the
They dive, they grab the back fin of the shark, they immobilize the shark, and then they attach the tag.
Okay, it's time to close the session. Thank you very much.
audience and a big round of applause to all the three speakers of the session.